Good morning, everybody. Welcome to the second session of the preparatory course. Last week, I tried to present these slides and I was having difficulties. So I thought I'll just mention them again and just remind you what and why the preparatory course is important. Today with us, we have certified clinical embryologist, Dr. Keshav, who will be giving a lecture as well. So you can obviously contact him to get your feedback as to how he felt it. And the question that comes to the mind is, what is preparatory course and how does it help me? Well, the simple answer is that preparatory course helps you to revise your basic understanding of cell biology and practical embryology skills, including troubleshooting. And I'll be giving a talk on that towards the end of the course. It also allows a better appreciation of regulatory bodies. Now in India, you are going to be having the ART bill, so you have to be prepared for it. Although you will not be questioned in the exams about the Indian ART bill, it's going to be more related to the EU directives. But if you look at the structure of the regulatory bodies and what they ask you to follow through are very similar. And that is the reason why it's very important to know and understand what is required in your country, i.e. in India. And why there's a need for regulations which allows you to put into place a very robust quality control management system, and its significance will also be discussed. And it prepares you for the Eshre certificate exams also. The other question that you may also want to ask yourself at this stage is, why should clinical embryologists do it? Well, there's a host of reasons, and there are some that I've listed here, and that include the demonstration of basic or advanced knowledge of clinical embryology, to obtain the most internationally recognized clinical embryology certificate, to acquire an international certificate formally recognized by Council for European Specialist Medical Assessment. What it also allows you to do is officially record your clinical embryologist status on the list of Eshre certified clinical embryologists. It allows you to participate in the continued professional development, the CPD system, and to receive an official printed annual CPD certificate demonstrating educational profession and scientific activities. Furthermore, to officially record your CPD status on the world map of Eshre certified and CPD activity clinical embryologists. And most importantly, for most of you, as you are looking forward to advance your careers, it will facilitate opportunity to practice internationally because you would have proven that you are capable and you have a certified, Eshre certified clinical embryology certificate. The curriculum vitae is quite long, so we have divided that into 5 different sessions. And as mentioned last time, instead of the 29th of November, the last day would be the 28th of November. After the lectures, there will be a final exam, and we'll talk about it later. These are all eminent faculty who are going to be presenting the talks to you. Most of them you would know, but those who you do not know, they will be very happy to discuss things with you, share things with you, and of the last session, 4 of them presented their talks. Dr. Sarabjit was one of the first certified embryologist, Eshre embryologist. Dr. Ane Sundan, whose baby this has been, who had the vision to introduce the certification for all the embryologists, is with us as one of our moderators. And Dr. Keshav Malhotra, who is going to be lecturing today, is also certified embryologist. And we are very pleased that we have a galaxy of faculty members who would be delivering their vast knowledge. All of them put together, we are looking at more than hundreds of years of experience. Today's talk is going to be presented by Dr. Pankaj Talwar, Dr. Keshav Malhotra, Sweta Mittal, and Dr. Surin Goman. At the request of Dr. Pankaj, we have changed the order and the development biology, which covering embryonic stem cells, the fetal ovary, the fetal testis and gamete interaction until first cleavage would be delivered as the last lecture today. But to start the talk, we'll have Dr. Keshav Malhotra talking about developmental biology, and he will talk about embryo development in implantation, followed by Dr. Sweta Mithal talking about post-implantation embryology, early pregnancy, and finally by Dr. Surveen, who will be talking about anatomy and function of the female reproductive systems and oogenesis. The delivery of the lectures, because of the time zones and some internet difficulties, we felt that we may deliver it either live or recorded. The next lecture by Dr. Keshav Malhotra will be delivered recorded, and where possible, the faculties have been given a choice to deliver it of the best they can think. Faculty will be presenting at the time of, they will be present, those lectures which are relayed as a recorded lectures, but the faculty have been requested to be present so that they can answer the question. Like last time, what we propose to do today is to actually have the question and answers towards the end of all the 4 lectures, a collective question and answer session. So what I would request to each and every one of you, and we had a good interaction last time, is to write down your questions and please keep on sending it to us so that we can compile them together as moderators behind the scene and have them ready to be answered by the panel. 3 moderators, Professor Ane Sundaran, Professor Kuldeep Jain, and myself, will try and say the course conducts well, and if there are any difficulties, please feel free to get in touch with us via email or this session. The additional reading material, some of you have asked for it, will be sent across by an email. I have asked the concept organizers to do that. But it's also available on the Eshre website, and if you are an Eshre and IFS member, there's excellent educational material freely available on their website. Briefly about the multiple choice questions. Last session, some of the candidates, 82 candidates sat the exams. There were a few who joined us late and also a few who have registered after that session. For those, we have decided that at the end of the session, after Q&A, we will run the question, the quiz for them for 30 minutes and give them the opportunity to understand what the questions would mean and how well they can do. The answers for the results will be all presented next week's session at the start, and it will be presented as histograms for each question, reflecting how many of you answered that particular question correctly and how many did not get the answer. No marks will be given for this trial one, but marks will be given for the final exams. The other question that was asked last time and have been approached, what are the requirements to take the exams? There are 2 exams that you can take, clinical embryologist or senior clinical embryologist. If you have the membership, you can get this information by clicking onto the Eshre section. Go onto the Eshre website and you'll get all these important requirements. Clinical embryologists must hold a BSc in natural or life sciences, whereas a senior clinical embryologist needs to hold a MSc or PhD in natural life sciences. Question also was, can MD candidates apply for it? And the answer is yes, they can. The only difference is that for the clinical embryologist, you have to have at least a 3 years hands-on experience with human gametes and embryos, whereas with senior clinical embryologists, you have to have at least 6 years of hands-on experience of human gametes. You need to submit a logbook, and I'll explain that to you next. And the logbook should have at least the following items listed: oocyte pickup, all the way down to thawing, warming of oocytes and embryos, semen analysis, semen preparation, and you need a minimum cases of 50 should be recorded. The way you would fill out the logbook is demonstrated here on the slide. You need to have a start date. You need to have a stop date. Even if you're still completing, you should enter still going on. Total number of clinical cycles performed by yourself since the start of your employment in this clinic, that clinic where you're working, and total number of clinical cycles performed in the clinic by yourself and others in 2020 only. It's important that you have a supervisor who signs for this logbook, and your name has to be there. Going back, when you're preparing your application for admission, a copy of the highest obtained diploma in natural sciences should be submitted. A BSc will be uploaded with your MSc or PhD. Official English translation of your diploma, academic transcripts, an official name change document if you've changed your name since you got your transcripts, a copy of your passport, and as I mentioned, the logbook. And there has to be 2 references different from your supervisors. And these 2 references can be your clinical lead, i.e. the medical director, or even your head of the unit can sign for that. And a resume in Europass format should be presented. And this is all described on the website for you to have a look at it. Any other additional information or for the courses is listed readily available on this website, which I'm presenting here. You will have access to this, and we will be more than happy to share this with you. And after submission of your application, should you have any questions, please contact certification@eshre.eu for them to come back to you, and they are very good in coming back. And the final slide is an announcement slide for today. As we mentioned, that some of the candidates were late joining us last week, and also there are new candidates who have joined. To give them a chance of understanding what an exam entails, we have decided for only those candidates, 82 who appeared last week will not be allowed because we'll block their entry, but only for those who haven't taken the exams last week, please remain behind and via chat, let us know who you are with your name so that we can do the needful at the end of the session. We'll run a trial at the end of today's session. And as I mentioned, the results will be presented next week as overall answers to each question. For final exam, you'll receive an individual mark, so you know how well you've done. 66% is a passing mark, and this is based on the curriculum circulated. We need the feedback from you on the material delivered by us. So there will be an appraisal form which will be sent out to you for you to fill out and complete so that we know how well we are doing, what we have missed out, and how we can improve on that. And this will be very important. And without anything further to do, I would now like to invite Dr. Kuldeep Jain, our chairperson for this course, to introduce our next speaker, and let's get the ball rolling. Thank you very much. Enjoy the day.
Thank you, Dr. Jayant, for all the information which has been actually been asked by every delegate, and I hope that now the things are very clear. And even if you are having any problem or any confusion, you can write to us on WhatsApp or on mail, and we will be happy to answer all your queries. So good afternoon, everyone. It is again a pleasure to invite all of you and welcome all of you to this second session of our preparatory course. Before we start, I will like to announce two more announcements. One is the repetition to make sure that all of you are available for the last session on 28th November instead of 29 November. That is the change which we have already announced last session also. And second thing is for IFS certification also, the requirement and eligibility are the same as Dr. Jayant has highlighted for Eshre certification. Only thing here, you have to be the member of IFS, but the rest of the eligibility are same. And when you complete and pass the exam, which will be conducted on 28th of November, you submit the logbook and the requirement of 3 years experience and the eligibility criteria, all you have to be fulfilled and submit before you leave the final session. So last date for submitting the logbook is extended to 22nd. Previously it was 15th. So now we have extended it to 22nd. So please submit your logbook by 22nd to IFS office. You can send it via mail along with your documents, as well as a covering letter from your supervisor. So that was the important announcement, which is very important for your certification. Starting today's session, I invite Dr. Kesav Malhotra, who is a upcoming embryologist, and as Dr. Jayant has already highlighted, that he is one of the Eshre certified embryologist. We are having almost 26 plus embryologists who are certified by Eshre in last 4-5 years. He is having a master degree in clinical embryology from Monash University, Australia, and he is the lab director currently at Rainbow IVF, and now the director of Malhotra Embryology Training Academy. He is running a lot of courses, and I will also like to attend some of these courses, Kesav. I heard that they are very, very good. So he will be speaking on the developmental embryology. So Kesav, floor is yours.
Thank you so much, sir, for that very kind introduction. I am facing issues with the internet, like Dr. Jain said. So we are going to be taking this lecture as a pre-recorded version, but I'm going to be there with all of you. So if you have any questions, please put them in the chat and I will be answering them, and I'll be there for the Q&A session. Rest, we can just start the lecture, Rajesh.
A very good evening, everyone. First of all, I'd really like to thank IFS for inviting me here to actually present to a platform which I feel is really, really important for our country, because standardization of services can only be achieved once there is an accreditation system for embryologists. And I think Eshre is one amazing platform where embryologists can actually get accredited by giving a certification exam, and IFS has been pushing this quite a bit, and I'm really grateful to the team to actually invite me to speak and actually teach a difficult topic. Even though it sounds simple, it's one of the fundamental topics of embryology, but it's still not easy. And today we're going to be talking about embryonic development from cleavage to implantation. And I'm going to be I'll touch upon a lot of different aspects. I'll try and touch upon the division patterns. I'll touch upon the polarity a bit, the kinetics, the metabolic changes that actually happen. And hopefully by the end of this lecture, we'll have a comprehensive understanding of how the embryo develops, and hopefully this topic is actually going to help you in the preparation for your exam. So without wasting much time, let's begin. Now, we all know that the fertilization process, which has already been discussed, which is followed by the embryonic development process and implantation itself, is a complicated procedure. It has a lot of steps which are involved. It actually requires a very different kind of regulation, which, if you look at natural conception, if you look at the physiology of embryonic development and embryonic implantation, you'll actually find out, okay, so you have the egg which is actually ovulating from the ovary. It's picked up from the fimbrial end. It has to traverse through the fallopian tube. So somewhere around the ampulla, it's actually going to get fertilized with the sperm. And you have to understand at this point that throughout this developmental phase, or throughout this passage through the fallopian tube, there are a lot of changes as far as the environment is concerned. There's so many changes which are happening as far as the regulation, the uptake of metabolites, the excretion of metabolites is concerned. So there's so many things that are happening together, which actually ends up in a beautiful blastocyst, which then comes into the uterine cavity and then has to adhere to the endometrium implant, and hopefully that embryo would result in a pregnancy and a live birth. I'm going to be talking about the kinetics of the embryo first, because let's get the easy stuff out of the way first. So now this is the Istanbul consensus, which really talks about embryo grading and also the development of the embryo. And based on this consensus system, we have the exact time points where we should be assessing the embryo. Now, based on this, you would want to do a fertilization check at around 16 to 18 hours post insemination, maybe a syngamy check at around 24 hours post insemination. An early cleavage happens at around 26 hours plus minus 1 for ICSI, about 28 hours plus minus 1 for IVF. A day 2 embryonic assessment happens at around 44 plus minus 1 hours, where you'll get a 4-cell embryo. A day 3 embryonic assessment happens at around 68 plus minus 1 hours, where you'll get an 8-cell embryo. Day 4, or the morular stage, is at 92 plus minus 2 hours, and a blastocyst, a well expanded blastocyst, should ideally be forming at around 116 plus minus 2 hours. So that's the kind of timeline that the embryo has to follow, and any deviation from this kind of a timeline, if it's going faster, if it's going slower, or if it's skipping a few developmental checkpoints, at those stages, we'll probably, it's the job of the embryologist to pinpoint these embryos and maybe deprioritize them for transfer. So you'll select an embryo which is actually following the correct timeline and transfer that first. And if you have no other option, you would want to then transfer an embryo which does not have that potential. And I'll talk a little more about this when I touch some of the info that time-lapse has actually brought as far as this parameter is concerned. Now, apart from the kinetics, you also would want to look at the cleavage patterns, and that's also something that we as embryologists really have to track. So there could be uneven or unusual cleavage patterns which could also exist. So the embryo can go directly from a 1-cell embryo to a 3-cell embryo, or it can become a 2-cell and then go directly to 5-cell or 6-cell. Or what can happen is that it goes from 2-cell to 5-cell, and it then comes back to 4-cell. And that's one of the negative factors of a normal morphological assessment, that you would not be able to track these intricate abnormalities which a time lapse actually helps you with. And there is a lot of data to suggest that if an embryo is not following the correct timeline, or if an embryo is not following the correct cleavage patterns, obviously the implantation rates for such embryos is poorer. So this is again data which shows abnormal syngamy. This is data which shows abnormal cleavage, and again the implantation rates for such embryos is poorer. Now, this is quite a complicated slide for a young embryologist to begin with, where you can actually see these different kinds of terminologies, which is T0, TF, TC, T1, T2, T3. It sounds complicated, but once you experience time-lapse imaging, and once you experience how an embryo really divides, then it'll become very easy for you to correlate with these timelines. So let's say that T1 is time to 1 cell, T2 is time to 2 cell, T3 is time to 3 cell, T4 is time to 4 cell, TM is time to morula, TB is time to blastulation, TEB is time to expanded blastocyst. So this is the kind of timeline that an embryo follows, and what we are now trying to assess is how long it's taking for an embryo to become, let's say, a 4 cell. How long does it take from an embryo to become from a 4 cell to become an 8 cell, from 8 cell to become a morula, from 8 cell to become a blastocyst? Now these are all the different kinetics that are being evaluated at the moment, and based on these kinetics there are certain softwares which can actually give you a scoring for the embryo. So let's say I'm going to be talking about one software which is known as KID score. KID basically stands for known implantation data, and here the embryo the scoring is divided into 1 to 5, and you have, like, let's say score 1, where which is the poorest, where the embryo was very fast to begin with. So the early divisions that were happening, time to like T2, T4, T8, even like T8, all those were accelerated. Then you have a score 2 where your initial divisions were a little slower. Then you have score 3 where it's irregular, but like there are increased divisions. Then score 4 or KID score 4, again irregular pattern, but again slower. And then you have a KID score 5, which basically follows the Istanbul consensus for embryonic development, and it will actually have the exact time points for embryonic development as such. So that's the kind of fascinating stuff that time lapse actually brings to an embryologist. And based on all of this, we can now actually really see the kinetics of the embryo. We can track each and every cell division, and there's so much to learn from these cell divisions, which hopefully the next few slides will appraise you all about. Now, the second thing that I really wanted to highlight here is basically your embryo polarity. Now, we know that mitosis, which is generally what's happening during cell division. So mitosis in blastomeres should actually be producing 2 equal sized daughter cells. And like when the division is asymmetric, one of the blastomeres is going to be bigger, one of them is going to be lesser. So one is going to inherit more cytoplasm, one is going to inherit less cytoplasm. And it can actually determine the fate of the embryo. It can determine the fate of what cells or what amount of cytoplasm is actually going to become the inner cell mass, what amount of cytoplasm is actually going to become the trophectoderm, etc., etc. So this brings us to the concept of embryo polarity. Now, I have to admit that this is a concept which was quite, like, in the late 90s and the early 2000s, this was gaining a lot of significance because people were actually trying to study like the kind of cytoplasm or the kind of divisions which are actually resulting in better quality embryos and resulting in better blastulation. And I think that now this has lost some of its significance because the newer like the students who are coming up now really don't have that much of an understanding of this kind of a phenomena, right? So let's understand that human oocytes are polarized right from the earlier stages of their formation. It consists of an animal pole and a vegetal pole, and this animal and vegetal gradient that is there in the cytoplasm of that even a single cell embryo, that basically, like, right from that point of time we have some sort of a understanding that okay, this is the part which is going to become the inner cell mass, and this is the path which is going to be the anembryonic pole, or it's actually going to become the trophectoderm. Right now, there are 2 sorts of divisions which can happen. First can be along the meridian, and second kind of division can be like around the equator. So if you look at the slide here, that's the equator. So that's the kind of, so if the embryo divides like that, it's an equatorial division. And that's the meridian. So the embryo can also divide like something like that. Now the first cleavage that really occurs is along the meridian. So if I look at just this blastomere right here, you can see that, okay, let's say that this is the animal pole and this is the vegetal pole. So you're going to have an equal division of both these kinds of cytoplasms, right? And then if you look at the other kind of division that happens, you have the animal pole here or the vegetal pole below, and the embryo separates like that. So it's going to solely differentiate one blastomere to become to form the animal pole, and the other part of the blastomere which is going to form the vegetal pole. So that's the significance of embryo polarity. I'm going to try and like maybe simplify it a bit for you. So again, like I was mentioning, the first division is along the meridian, and when it's along the meridian, you're going to have an equal split of the animal and vegetal poles into both the blastomeres. Now the second division can be of various kinds. So the most commonest and the best kind of division is when one blastomere divides along the meridian, the other one divides around the equator, and that should be giving you a tetrahedral kind of an arrangement inside the zona pellucida, and that's how like a pyramid shaped kind of a arrangement of the blastomeres, and that's the best kind of arrangement that you would get. And if I look at the older studies here, these are the kind of embryos which would result in better kind of blastocysts and better implantation rates. Now, the second sort of division would be when both the blastomeres are then like dividing along the meridian itself. Now, when this happens, you're going to get like clover shaped, or you're going to get like a flat sort of an arrangement where you will see that all four blastomeres are just compactly stuck within the zona pellucida, and I have images of embryos which show this, and that's the kind of arrangement that you would see. And again, similar sort of an arrangement can also happen if both the blastomeres are dividing along the equatorial axis. And again, like I mentioned, what we're going to basically get from looking at all of these different kind of divisions, especially the early divisions, is that you'll be able to predict which embryo is going to form a better blastocyst, which embryo has a little more potential as compared to the others, and that's the only significance that you will get in a certain scenario. And we do have contradictory evidence to this as well, which I'm going to touch upon just after a few slides. So like I was mentioning, these are the different kinds of embryos that you would see. These are examples of all the published work, which basically concentrates on the possible allocation of like a single founder blastomere for the trophectoderm and like the inner cell mass as well. So these are papers which were basically studying whether the inner cell mass actually arises from a single blastomere, or it has a more differentiated approach where different sorts of cells can actually get together and form the inner cell mass or the trophectoderm, or whether the fate of the cell is known right from the start, or does it actually happen later on, right? And at this stage, if I talk about a little bit of like MCQ questions for all of you, this is mainly governed by like the presence of maternal leptin, the presence of STAT3, and there are other different maternal proteins which are positioned at the animal pole, which actually determines this particular concept. So again, I'm just going to go through this concept very quickly. So you can see that you have a 2 cell here, and based on that, you have that polarity, and this main polarity, like this polarity of blastomeres is generally governed by the polar bodies which are present within the oocyte. So that also gives us a certain idea, and then based on like you follow these divisions up, and you should be actually you should be able to track the part of the embryo which is becoming the embryonic pole, which is the inner cell mass, and the part of the embryo which is going to become the anembryonic pole, which is opposite to the inner cell mass. And that's pretty much it. And again, now I'm going to talk a little bit about the new things that are coming up. And so this is courtesy of Dr. Tanaya, uh, and this is actually her PhD thesis. And basically, what they were trying to study was, again, they were trying to study the distribution of leptin and STAT3 within the oocyte and how it's actually affecting the polarity. And also at this point, they're looking at OCT4 expressions, and they're looking at beta HCG transcripts, especially at the 4-cell stage, to try and figure out whether this fate of the embryo is determined early on. And like it it they could say, like, based on this study, that the early interpretation of like Robert Edwards' model of this fate and repositioning of the blastomeres could be challenged. But again, we still don't know whether this polarity actually affects development, this polarity actually has an effect on implantation potential, or whether the implantation potential of non-tetrahedral shaped embryos at 4-cell could actually affect the development of the embryo, could actually affect the fate of the embryo. This thesis really challenges that. Okay, let's move on to the metabolism of the embryo. And this is also very fascinating because, the first slide that I showed you, where you have the oocyte which is getting ovulated, and then it's picked up by the fimbria, and then it has to pass through the fallopian tube and then come into the uterus. Now, most of these studies, which actually found out what the embryo was actually taking up, have actually been studies where they've taken secretions of the fluid from different parts of the fallopian tube, and actually looked at the presence or the of different kinds of metabolites which are actually present there. And based on these findings, your culture media, especially the sequential culture media, were actually developed. So this is how fascinating this particular subset or particular like branch of or niche of embryology is. So let's look at the metabolism right from day 0. So this is when the egg is actually ovulated, and we have to understand that the egg is quite a quiescent structure. And what I mean by quiescent is that you have the same egg which is present within the ovary for so many years before it actually gets selected, before it actually gets ovulated, and then is coming into the female reproductive tract. So looking at like this particular logic, it's safe to say that the metabolism of the egg is not that much. It doesn't require much, and it's quite basic. So when I say it's quite basic, I just want to emphasize here is that the egg does not have the capability to actually metabolize that much, and therefore it requires a lower source of energy, and that energy really comes from the pyruvate. Now, if I provide an egg with glucose, it cannot utilize it because it does not have glycolysis, which happens within its cytoplasm, and therefore you basically need pyruvate as the primary source of energy at this point. The other thing that we also want to emphasize here is that at this stage, it is still susceptible to DNA damage, and most of this DNA damage occurs because of the presence of high oxygen within the culture system. Now, again, I'm going to go back to physiology and go back to the uterus and the fallopian tubes. Now, both the fallopian tubes as well as the uterine environment is highly anaerobic, and again, here the oxygen concentrations are going to be quite low, and that is what we want to try and mimic when we actually put these embryos in vitro culture. So you would want to maintain low oxygen, and that's going to actually produce the best quality embryos from such an egg. And again, at this point, if I talk a little bit about amino acids, glycine and taurine are required at this point for osmoregulation and pH stability, and also glutamine for nucleotide synthesis. That's something that's required. Now, post fertilization, you have like the start of metabolism which starts happening within this particular zygote and the subsequent embryo which is going to form. And again, at this stage, the egg is still dependent on pyruvate. Maybe lactate can be supplemented here, because again, lactate can be easily broken down into pyruvate, so it's an easy metabolite, and that's also something that uh, is generally present in media, is also present in the secretions of the fallopian tubes as well. So that's something that exists till your 4 cell stage, or even till your start of the 8 cell stage, or start of day 3, where you will have the same pyruvate as the prime source of energy which is required. And again, here, if you look at if you compare the oocyte RNA and DNA status to a day 3 RNA DNA status, it's almost 8 times of the oocyte. Now, ATP production during this point of time actually occurs through the OX-FOS pathway within the mitochondria of this growing embryo. And again, glucose is something that is really not required at this point. And there have been studies, especially studies which were done early on in the 90s, which have said that glucose of more than 3 millimole has been detrimental to embryonic development at this particular point. However, a small amount of glucose, like let's say 0.5 millimoles, was shown to actually give better results. So that's something that's just a small piece of information for all of you. Now, after day 3, or after that 8 cell stage, when the embryonic genome actually switches on, that's where most of the like metabolism really starts to happen. So now it's the egg which is going from one of the least active metabolic cell within the body to a blastocyst, which is one of the most highly active metabolic cells or metabolic structures within the body. So that's the kind of transition that happens, and it's a good, or it's safe to say that the activation of the embryonic genome, which happens at day 3, is that turning point in this series of events, right? So now the embryo is switching from a catabolic state, where it was metabolizing the stored nutrients within its own cytoplasm, to an anabolic state where there is like rapid mitosis which is happening. There is rapid synthesis of new proteins which is going to happen. You're going to get new nucleic acids which are going to form, and then obviously the embryonic genome is switching on. So now, just by looking at this sort of information, you would obviously say that at this point you would want more energy, and more ATP has to be produced. And that is where your anaerobic glycolysis actually begins, and that is where you really require glucose as one of the prime sources of energy for such an embryo, right? And again, if you look at the other sorts of requirements for culture conditions, as far as this kind of a development is concerned, again, we want anaerobic culture, where we want low oxygen conditions. And again, this is something that most of us will maintain, where a 5% oxygen concentration is actually going to give you a better blastulation rate within your laboratory than a 20% oxygen culture condition. Now, at this point, this blastocyst, or this growing embryo, also requires non-essential amino acids and essential amino acids, which are really help in like biosynthesis and regulation of the enzymes and the metabolites which are being produced within this embryo. Now, when I talk about the the metabolites which we just referred to, this embryo, or let's say this blastocyst, is uptaking certain metabolites and excreting certain metabolites. And this is basically an interpretation from Professor Gardner's slides, where you have the blastocyst taking up glucose, pyruvate, amino acids, other sugars, oxygen, etc., from within the culture media, and metabolizing it and secreting or excreting lactate, ammonia, LDH, other peptides and factors, etc. So now, based on these metabolites, you can actually study the uptake of, like, let's say, glucose, or the uptake of oxygen, or the production of ammonia or LDH, etc., and determine whether that embryo has a high implantation rate or a low implantation rate. And these are some of Professor Gardner's works, which I have mentioned here. So this is basically the normal uptake of glucose, pyruvate, and excretion of ammonia. Here you can see that the glucose uptake, when it was between, like, let's say, 70 to 100, that the embryo actually became pregnant, or resulted in a pregnancy. When it was lesser than 60, it did not result in a pregnancy. You can actually also look at the different amino acids, and again, their turnover can also predict pregnancy rates. You can also look at oxygen consumption rates, and that can also, like, tell you a little bit about the uh, pregnancy and implantation rates. Now, whenever you don't offer the right kind of culture conditions, or whenever, even within the body, if there are hormonal imbalances, or if the conditions are not conducive, it's going to produce a lot of stress on the embryo. And this stress is mainly going to actually activate apoptosis. So let's say I'm culturing an embryo within my laboratory, and my culture conditions are not great, or the kind of culture media that I'm using is not the best. And what I'll notice in such a scenario is that the embryo, post, like, let's say, day one, or even like day two, you're going to get fragmentation which starts occurring. And these fragments which are forming all have been actually associated with cell apoptosis as well. So that's also something that we have to be on a lookout for. And I've actually mentioned some of these signals, regulators, and effectors for apoptosis, which could be one of your MCQ questions to follow on, especially in the examination. This is a study which actually tells us about what I was just talking about, where they actually used those annexin 5 or annexin V like stains, which is basically for apoptosis, and you must be like aware of it from its use in MAX, which is magnetic activated cell sorting. So it's the same kind of stain which is being used here. And they found out that when you had a fragmented embryo, that was actually staining for all of these receptors, which are positive for apoptosis. So that also tells us that the embryo viability of a fragmented embryo can be compromised. And what was the basic effect of these stressors on the embryo? Obviously, you have potential short-term responses where it could induce epigenetic modifications. There could be metabolic stress. There could be different kinds of gene expressions which can happen. And I was listening to Professor Gardner again speak just today on his talk on lactate and its use and it's actually its role in determining the fate of the blastocyst. And there was this good term which is known as metabologenomics, and that was something which is fascinating, because it's also these metabolites which can alter gene expressions, and that's something that we should be on a lookout for. And again, all of this will result in apoptosis. Now, if the embryo survives the potential short-term responses, it could also result in a lot of different long-term consequences, like an unbalanced fetal and placental allocation, altered maternal nutrition, abnormal fetal growth, abnormal birth weight, postnatal growth, cardiovascular and metabolic syndromes as well. So we have to be very certain as embryologists to provide the best on the most conducive environment for the embryo to grow. Now, moving on to the last part of this lecture, and that's going to be a little bit about how the embryo implants. So let's look at, basically, this is an embryo which is hatching, and this is a hatch blastocyst, and they've actually cultured it till day 10 of embryonic development. We know that research permits us to culture till about day 14, but this has been cultured on endometrial growth plates or endometrial cell culture lines, and you can actually see how the embryo like divides, how it then like changes its pattern. The cells, you can see that the cells are compacting, and then is going to just spread all over the embryo culture line, and that's when the adhesion and the invasion starts to happen. Now, for all of this to happen, the embryo has to first hatch out of its shell, and that shell is the zona pellucida. Now, this is a fantastic study which I could find from India, and this is Dr. Gunashila's group early on, and they actually studied the mechanics of human blastocysts hatching in vitro, and it was actually shown that at the point of the zona breaking, or where the zona actually ruptures from, there were these certain trophectoderm cells which exhibited a different kind of a morphology, and they were labeled as plump zona breaker trophoblast cells. And this basically uh, actually generated a phenomena where people thought that there are certain cells within the trophectoderm itself which were programmed to cause hatching. But again, we still don't know whether this is true for human embryos or not. In mouse, if I look at the hatching process itself, it's really almost predestined, where the blastocyst hatches at a certain angle from the inner cell mass. In humans, sorry about that. In humans, what happens is that it generally hatches right opposite to the inner cell mass, and that's something which is most commonly seen. This is how this. Let's just look at how an embryo basically hatches. Okay, I'm having troubles with the video, but I'm sure all of you have already seen an embryo hatch. So what will happen is that once an embryo is growing, you look at the zona pellucida and you look at the thickness of the zona pellucida, and around like 115 hours, what you would see is that the thickness of the zona pellucida has actually become half of what it was at the start of blastulation, and this is where you would call it as an expanded blastocyst. And again, after a few hours pass, you will start to notice that there are certain trophectoderm cells which start protruding from one point. Now you can clearly see it within this image itself, where just opposite to the inner cell mass you have the hatching process which has been initiated. And after that, if you keep following this embryo, you will see that this gap actually expands, and almost half of the embryo is actually going to protrude from it, and then slowly and steadily the remaining part also just squeezes and comes out of the shell that is actually present there. And this shrinkage of the blastocyst and then re-expansion also facilitates the hatching process itself. Now again, a certain few MCQ questions which could arise from this particular topic for all of you would be the role of cytokines in the process of hatching itself. And you can actually see that there are pro-inflammatory factors and there are anti-inflammatory factors which are causing an effect on hatching, where you can see that interleukin 1, interleukin 6, LIF, GM-CSF, tumor necrosis factor alpha, etc., are the pro-inflammatory ones. And then you have IL-10, IL-11, CSF1, and TGF-beta, which are the anti-inflammatory ones. And you actually, this is a nice paper. Again, this is coming from India itself, where they looked at the effect of the anti-inflammatory factors and the pro-inflammatory cytokines, and they found out that during the hatching process, it's tipped a little towards the anti-inflammatory ones, and during the adhesion and the implantation process, it's tipped a little towards the pro-inflammatory ones. So this is also a fascinating concept which actually really governs implantation. Now, when I talk about implantation, that's one thing that eludes all of us all the time, because we really don't have a very good understanding of embryonic implantation, because it's something which is not just determined by the embryo itself. There are so many other factors which are actually playing a part in the process of implantation, where it's the viability of the embryo, the quality of the embryo, especially the quality of the trophectoderm cells. It's also the receptivity and the selectivity of the endometrium, and it's also a crosstalk between this embryo as well as the endometrium. And all of these factors actually really govern the process of implantation. Now, we know that this process of implantation has different steps, where you have like apposition of the blastocyst, you have adhesion of the blastocyst, you have invasion of the blastocyst within the endometrial lining, and that is how the basically your embryo implants. And again, this is a fantastic paper by Deepak Modi, which I would really like all of you to actually go through, because it really talks about this battle between the endometrium and the embryo. Because naturally, if you look at physiology again, this is a foreign cell which is invading into your body, and now it's the maternal environment which really has to govern whether this foreign element is actually accepted, and then it grows into a fetus, or it's rejected. And that's the basic concept of implantation. Now, this basic concept, like I told you, there are so many factors which govern hatching. There are so many factors which actually govern implantation as well. And again, just to name a few of these, you have all the interleukins. You have interleukin 1 beta, you have interleukin 6, you have IGF2, you have EGF, you have VEGF, you have GMCSF, you have TGF beta. There are so many things which can actually really affect the implantation process itself. Also, COX2 and the prostaglandin pathways are also involved here. So again, this is a slide which basically summarizes all of that. Now, just a small part about the receptivity and selectivity. Now, if you notice our species and the reproductive, like human reproduction as such, it's actually characterized by a high wastage of embryos. Now, most of the embryos that are formed are actually rejected, and they don't really implant, and that's one of the things which is quite like specific to humans as such. Now, part of it could be actually correlated to the endometrium as such, because like we know that obviously the endometrium has to be receptive to receive the blastocyst, but now we're coming to a new concept which actually says that the endometrium actually acts like a biosensor, and it's more of the selectivity of the endometrium which like governs implantation. So again, this is again from Deepak's slides, but you have to actually achieve this balance between receptivity and selectivity in order to actually achieve implantation. Now, I'll just give you a small example of how this works. Now, there are studies which say that if you're transferring a good quality embryo with a bad quality embryo, it's both of these embryos don't implant. And when you compare it with just transferring a single good quality blastocyst, obviously the implantation rates are higher. Now, what's happening here is that because of the endometrium sensing that there is a poor quality embryo within the environment, it just becomes non-selective, and again just discards the implantation process itself. And this is the phenomena which is emerging, and a lot of fascinating research is happening on endometrial selectivity at this current point of time. So with this, I'm just going to end with this nice video which is actually from Nature, where you can actually look at the day 6 embryo. So the green cells, or the green stained cells that you see at this point, is going to form the inner cell mass. The blue ones are going to become the trophectoderm. And now what you look at is around, this is like day 8, where you see that the embryo has changed its like uh, the formation of the blastocyst itself. So you have the endometrium, or you have the trophectoderm, which is lying close, and this would be in close conjunction with the endometrium, and the inner cell mass lies on top. What happens after this is that the invasion process starts, and you have these red small vessels which start forming within these small cell gaps which are present within this blastocyst, or within this invading blastocyst, and that's where the new blood vessels which are going to come from the maternal side are actually going to be present there. And that's a day 13 blastocyst. That's how it looks like. And again, this is where basic research actually stops, because at this point, we're not allowed to culture embryos post day 14 in an in vitro environment. But just by looking at all of this, there are so many things that we can decipher. We can actually see the whole process of the invasion. We can see how the trophectoderm is and the inner cell mass is aligning, and what are the positions of both these cell lines. And it's an absolutely fascinating thing. And I think implantation is something which is probably the most fascinating concept as far as embryology and reproductive biology is concerned. So with this, I'd like to thank all of you for your patient listening. Hope this lecture was informative to you, and hopefully is going to help you with your exams. A special thanks to IFS for actually inviting me to present this lecture to all of you, and also thanks to Cooper Surgicals for sponsoring and supporting this initiative. It's very, very important that companies come forward and support standardization of practices for embryologists. And if you have any questions, I will be happy to answer. And you can actually email your questions to me at dr.keshavmalhotra@gmail.com, or get in touch with me on any of the social platforms, and I'll be happy to answer your queries. Thank you so much, and hope you have a great day ahead.
Thank you, Keshav, for a wonderful lecture. I think, yeah, as you said in your sum up, that it is really fascinating. Yeah, it is fascinating. Whole embryology, if you see, starting from the pickup, when you start seeing the COC, and then looking at the cleavage and then blastocyst. As much as you see, you try to get more of it. So I think, Dr. Jayant, we will be taking questions later, because they are not many questions at present. But I hope that we will have a good question answer session at the end. So Keshav, just hang around
I'll be here.
and join at the end. Yeah. Thank you so much.
Thank you very much, Dr. Keshav. Very enjoyable talk, and I'm sure that the candidates would have learned a lot. If I invite Dr. Arne now to introduce our next speaker, Dr. Sweta Mittal. Rajesh, has Dr. Arne got Dr. Sweta's CV?
Oh,
I
have it. I get.
Yeah, Rajesh will just project it. Yeah.
Thank you.
There we go.
Yeah, Dr. Svetlov is going to take us the next step in this fascinating story. Post-implantation. She's a senior consultant center at human reproduction at Sir Ganga Ram Hospital in Delhi. She's an MD. She's have had the Young Scientist Award, the Foxy Ethicon Travel Fellowship. She credited the first pregnancy from frozen thawed oocytes in North India. Then we were on the same spot, but I'm in a different country. It's nice to see. I've seen you so many times being in India. So she's now a present CEO convener, research and methodology specialist in interest group in IFS. And she's a past editor on IFS. Please, Dr. Sveta, take us the next step in this story.
Thank you very much. Rajesh, can you stop the screen sharing? Yeah, just a second. Thank you very much for such a lovely and kind introduction. Straight on, moving on to the next topic. Well, you've heard a lot in detail about the process of implantation. Let's move on and let's start imagining that what happens once the embryo has implanted and what is the process it goes through, how does the organs form, and what happens in early pregnancy or very early pregnancy failure. So I would not be completely discussing organogenesis, but I would be discussing the beginning part of the pregnancy. So if you have gone through that, implantation is a very highly organized process involving an interaction between a receptive uterus and a competent blastocyst. Till date, even if the time lapse has come in, but we really don't know what happens to these embryos once it's inside the uterus. It's like a blind room or a dark room which it is into. So the natural fecundability of a human is about 30%, and about two-thirds pregnancies are lost actually due to implantation failure. So the clinical pregnancies, what we see is just the tip of the iceberg. The basic three processes of implantation are embryo attachment to the endometrial surface. Now, the embryo has to actually have the cell receptors in such a way that it is directed to that part of the uterine environment which will actually accept this embryo, and it happens during the window of implantation. The moment these receptors and the sites are recognized, the embryo will implant itself. It will quickly start invading into the epithelium, which will start after the hatching of the embryo, that is from day 6. And then the first event what will happen is that it is the establishment of the placenta, because now the embryo has to, gain a good blood supply into the maternal system for it to survive further. So if we divide the stages of human development, we will be going through fertilization, pre-embryonic, embryonic, and the fetal stage. When does this whole process start? It starts as early as the second week of development. So implantation of blastocyst is actually completed during the second week of embryonic development. And at this time, the blastocyst undergoes morphological changes to produce a bilaminar embryonic disc. It might sound a bit new to you, but I will explain you that what is a bilaminar embryonic disc in the next few slides, giving rise to the germ layers. So now, till now, we know that you are aware that blastocyst basically consists of three things: the outer layer, which is the trophoblastic cells, the inner cell mass, and a blastocele cavity. So please remember these three important organs, and how the differentiation will start happening in the inner cell mass and in the trophoblastic cells, we will just see in a little while. So the bilaminar embryonic disc then will give to the ultimate germ layers which the baby needs to form all its organs. So if you can see the first picture, it is just showing that it is invading. The second picture, it has already invaded, and can you see the blood vessels which are all around the pools of them? And the third one is showing a further development of the embryo and the formation of that bilaminar disc. So this is, I would say, the most important picture, and please start going through this picture with me one by one. I will put my arrow on the first picture and I will explain, because if this picture is memorized in your mind like a photograph, you will be able to understand the rest of the part of my presentation. So the first one, actually the first picture shows that there is, two pronuclei. So this happens after fertilization, and this is what Dr. Keshav was telling, that after you do a normal fertilization or ICSI, what you see after about 17-18 hours is the pronuclei formation. The second picture is just a cleavage stage embryo, which is showing blastomere. The third picture is again compaction, so possibly this is morula, third and fourth. And the fifth is what you see in a blastocyst. What I said, the outer layer is a zona pellucida. Then you have the outer layer, yellow one is the trophoblastic cells. The red cells is the inner cell mass, and the yellow, light pale yellow, is the blastocele fluid. Now come to picture number 6, which says that now this has hatched. So typically on day 6, you will see that the zona has thinned out and the embryo has started hatching. The 7th picture now you see is that the embryo has found a place along the endometrium which is most receptive, and how I would say somebody eating cheese is exactly how, these cells have started burrowing inside the endometrial cells. By day 8, the cells, the whole, embryonic cells are completely, inside. They have crossed the endometrium, and now a barrier will come that it will further prevent its invasion. Otherwise, they would be placenta accreta, or they could be placenta prakreta. So it will get stabilized there in that particular place, and it will start growing. Now, at this picture, that is picture number 8, can you see now the inner cell mass? You can see a light pink color cells and a brown color cells. They have divided into a bilayer disc. Please remember these two terms. The bilayer disc, the upper one is called epiblast and the lower one is called hypoblast. And everything would be happening now henceforth from the inner cell mass with these two types of cells. So now when you go to picture number 9, you will see that the pink cells, which is the epiblast, has formed like a ring and has a cavity which is enclosed inside. And remember that this would form the amniotic cavity. So the amnion, which is the layer of the, a layer which covers the whole, fetus, which is like a shock absorber, which gives, the nutrition, is amniotic fluid and derived from epiblastic cells. The lower one, the brown one, now this will just form yolk sac and eventually this will vanish. Come to picture number 10. Picture number 10 now says that eventually this bilayer disc, if you can see pink above, brown below, and a layer of red cells in the middle, has formed a 3-layer disc. And this 3-layer disc is important because all our body organs will be derived from these 3 layers, which we will see. If you go to picture number 11, you will see another picture where now the amniotic cavity is surrounded by a layer of cells. The yolk sac is there. It will form a secondary yolk sac. And in the last picture, you will appreciate that how the folding of the embryo has started. So now it is no longer a spherical disc because embryo has to have a cranial end and the fetus has to have a caudal end, and so there will be an embryo folding, and this is how the development of fetus will happen. So if you can picturize the epiblast, the hypoblast, if you can picturize the double layer cells, if you can picturize a triple disc forming, if you can picturize the amniotic cavity, and if you can picturize how the embryo has folded, I think half my job is done. So now what happens? By second week after the implantation, the implanted embryo becomes more deeply embedded in the endometrium. So you can picturize the picture I said. Further development of trophoblast into placenta, which is very important, and development of a bilayer embryo, amniotic cavity, and yolk sac, which we have seen in the pictures. So trophoblastic cells. Now the simple trophoblastic cells will divide into 2 layers. One is a syncytial trophoblast and one is a cytotrophoblast, and we will see in the pictures. Now can you follow? You can see the embryo, you can see the blue cells, which are the epiblastic cells, which forms the amniotic cavity. The yellow cells are the hypoblast, and if you see the light green cells and the dark green cells, so the light green cells are cytotrophoblast and the dark green cells are syncytotrophoblast, and this red are the enlarged blood vessels which are from the maternal side. So by day 9, what happens is the trophoblastic tissue is further differentiating. It invades the maternal tissues. The first thing is that there is a lot of stem cell population because the cytoblast is actively proliferating. What is happening to the inner cell mass? Which we know it has divided into epiblast and hypoblast. The epiblast will contribute to the formation of amniotic membrane and the amniotic cavity, which will be like the most important cavity for the fetus, and a hypoblast which will be just forming the yolk sac, and then that will not serve much of a purpose later. So this is what has happened by day 9. Go to the next picture. By day 12, can you see syncytial trophoblast has established so much of connection with the maternal blood cells? So syncytial trophoblast is the one which would be invading the syncytium, which is derived from the cytotrophoblast, and more vascular channel would be formed, and after all, later on, it will form a complete placenta with a feto-maternal, I would say, transfer of nutrients, which would be happening through this organ. And the last is by day 13, that the trophoblast, now can you see the whole of embryo is now surrounded with the forming placenta, which is very, very vascular. It differentiates, invades the further into the maternal tissues. Now it will break all the maternal capillaries, because you know what will happen. See, it is the maternal blood which is flowing in like in a sinusoidal pattern, not as how we see in our own arteries. So this will be like it will be like a smooth flow through the maternal vessels, and all the fetal vessels tissue would be surrounding this maternal sinusoids, and it would be deriving the nutrition. So the trophoblastic lacunae which fill with the maternal blood, and this is the final thing what has to be achieved. What happens to the inner cell mass? The same. It has just divided into two layers, which we can see in this picture. So I hope that this is clear, because these are very theoretical lectures, and if you do not have a picturization of them, then it becomes very difficult for you to understand. Now moving on further, this is just a lateral view. So just to emphasize, you can see the blue is the epiblast and yellow is the hypoblast. We have the amniotic cavity on the top. We can see the trophoblastic cells, the light green and the dark green, and how well the sinuses have been formed. Now, what I want you to picturize further, that why I have brought in this picture again, for you to have one more recap. Look at picture number 10. In the picture number 10, you can see that there is a central depression now. So earlier, you could see that in picture number 9, the cells were straight. In picture number 9, the epiblastic cells and the hypoblastic cells were absolutely straight. But further on, you would see by day 14 that there is a bit of a dent which has come in the center, and you will wonder that why this dent has come, and I'll be telling you very soon that that is also very important phenomena. So in embryology, every little change what is brought in the embryo and later on in the fetus has a significance, and has its own importance. And then we just wanted to show that how the amniotic sac grows on day 12, in the 12th picture, and how the folding of the embryo takes place. So I will bring you to this depression again. So coming to this word, gastrulation. Gastrulation means it is transformation of the blastocyst into the gastrula with primary germ layers. To be putting it in a very simple term, conversion of bilayer disc into the 3 germ layers, which will be called ectoderm, mesoderm, and endoderm, by the rearrangement of cell, is called gastrulation. So basically what it involves a lot of cell movement. This will help in attaining the new shape, because we know that blastocyst was a spherical structure. Now we have to form a fetus, so it has to change its structure and morphology of the embryo, and from bilayer epiblast it forms into a triple layer disc. So at 2 to 2.5 weeks embryo, it is only 1/10 inch long, and a groove forms. So now can you see the top layer? And there is a groove which is called the primitive streak. A primitive streak is nothing but it is like a collection of cells which happens, and it forms a depression. It forms a depression, and if you can see that the uppermost layer is the ectodermal layer, and there is a primitive streak which has formed, and this primitive streak is causing a depression in the cells. The cells will migrate into this streak, and that is how between the ectoderm and endoderm, between the epiblast and hypoblast, because epiblast will form ectoderm and hypoblast will form endoderm. So how will the middle layer come? So the middle layer will come because of this primitive streak, because this primitive streak is causing a depression in the ectoderm and making certain cells migrate, and these cells will migrate between the ectoderm and endoderm, and these cells will be called mesodermal cells. So if you can see the migrating cells in this picture in the middle, they will be called the mesodermal cells. So this is a picture of the primitive streak. Can you see a black line? This primitive streak. So but why is this primitive streak required? It is a transient thickening. But why? Because number one, it will give rise to notochord. Notochord is the spinal canal. So it is required for that. It will give rise to mesoderm. Mesoderm is one of the most important cells, because it is contributing to majority part of the body. It is required for the formation of the dorsal face. at the caudal end. So at the caudal end, not at the brain end, from possibly somewhere around the neck. Most important that when humans have a bilateral symmetry, this is because of this central depression, what is caused by notochord. So this is important. Otherwise, we would not be symmetrical in nature. And then the start of triple layer, and that is why it is important. So please remember what is a primitive streak and what is the importance of primitive streak in us. So in gastrulation, it is just again showing a primitive streak. It is causing a depression. It is causing migration of cells. And you can see that a definitive mesoderm and a primitive endoderm is changed to a definitive endoderm. This picture is just showing migration of cells from inside. It is moving towards upward in a semicircular manner. And this is just, I will not explain any further because I've already explained that how the migration of cells happens and how our body is converted into 3 layers. The outer, the dorsalmost layer is called ectoderm. The middle layer is called mesoderm and the inner layer is called endoderm. Now, there are many structures which can be, arising. I think instead of that, you can read theoretically. But if you are made to remember important 3 structures which would be derived from ectoderm, then you cannot forget the nervous system. So brain, your spinal canal, your spinal nerves are all from ectoderm. The skin, which is epidermis, and pituitary gland. And there are many things like cornea of the eye, which you can just see. But the important 3 things to remember is all our nervous system is derived from the outer layer of the embryo, and that is called ectoderm. Skin and pituitary gland. What about mesoderm? Mesoderm contributes to the whole of skeletal and muscular system, our whole of circulatory system. A part of urinary bladder and reproductive system is derived from mesoderm, and endoderm gives, so endoderm means everything with the canal, digestive system, our respiratory system, and portions of urinary bladder and reproductive system. So if you really have to remember, just remember these gross, organs which are developed from all the 3 layers, and that would be good enough. Another important aspect when I'm talking of ectoderm and formation of the spinal canal is that organogenesis starts from day 16 of implantation. Now follow this picture first. What is the first organ system to be formed in the body? Which is our nervous system. So if you can see the purple plate, you can appreciate that there is a purple plate with the central depression. Now this depression is accentuated. You can see in picture 2, and you can see that both the green bands and the blue bands are coming closer to each other. And this is what is called that transformation from a neural plate. This ectodermal plate is now a neural plate to a groove, which is called a neural groove. You can see that how the neural groove completely gets detached from the blue plate, because this will form the epidermis, the skin, the lateral part of the ectoderm, and the whole tube will form, actually the neural tube will form the spinal canal. So this is very important that our whole spine, the spinal cord, is formed from the neural tube. So this process of folding around the primitive streak upwards to form a closed tube along the whole length of the embryo from cranial portion, which forms the brain, and the caudal portion, which forms the spinal canal, this process is called neurulation, where the neural plate gets converted into a tube-like structure, and this is the beginning of the formation of nervous system. Now, around the same time, this is just a picture which is showing that how the embryo gets folded and how the fetus is formed, and from cranial to caudal portion, cranial will form the head and the caudal will form the end of the back and the limbs. Next, at the same time, is the formation of heart. Now, I will not be going into the details. We can detect the cardiac activity in a fetus from 5.5 weeks of gestation. A simple tubular heart is pumping blood from the placenta through the umbilical cord and bringing oxygen and other nutrients and returning the waste to placenta. So this is a picture which is showing that how it develops into 2 tubular structures. It comes together, it joins, then forms is the 4 chambers. So at birth, the child will have the four chambers and the foramen ovale will close, because during intrauterine life, the fetus is not breathing, so there is no pulmonary circulation. There is a direct circulation which bypasses the fetal liver and just goes to the placenta. So the whole exchange of oxygen in the fetus is not happening through the lungs, but is happening through the placental circulation. And that is why ductus venosus and your foramen ovale will close after birth, so as to establish the normal fetal circulation. Now, sometimes you would be hearing these words, what are somites? Now, look at this picture again. You can now appreciate that in the center is the spinal canal. Now, from the mesoderm, so mesoderm was what? It was a cluster of cells. Now, these cluster of cells will actually become like small blocks, okay? Small paired blocks. So, you will have same number of blocks, which is from the paraaxial mesoderm. That means from the side of the mesoderm. In the embryonic stage itself, they will form about 42 to 44 in pairs, but some of them they degenerate. Why it is important? Now, this is important for segmentation, because see, if we remember, our body is formed in segment. So your vertebrae, you know that there are so many vertebrae, ribs, multiple in number, spinal nerves, they will be arising between the vertebrae. All the trunk muscles and our cartilage are from the somite, which are derived from the paraaxial mesoderm. So can you see these blocks which are by the side of the spinal canal, and these are called somites. So another term which you would be remembering are somites. So just to have a complete, picture, so 3 discs, notochord forms in the center. This notochord is important in the mesoderm because then this is the beginning of the spinal canal. The neural tube will, instead of a plate, will now getting curved. It's dipping inside. It has curved more. You can see that the mesoderm has moved below the spinal canal. It has moved to the sides because now it is forming somites, and from there the segmentation will happen. And the last is that the complete neural tube has formed. It will form brain and the spinal canal, and so on and so forth. Important to remember, amnion, just recollect, it was formed from the epiblastic cells. It surrounds the embryo. It contains bag of water. It acts like a shock absorber. Chorion is the principal part of placenta formed from the trophoblastic cells. Metamorphosis is something which is differentiation, which starts from about 6 weeks of gestation, where the embryo differentiates into legs and arms, formation of facial features, sensory organ development, and pigmentation of eye. From the 8th week, now the embryo is called fetus. This is how the circulation happens. From the 2nd month of gestation, there is a very rapid growth happening, fine-tuning of systems. So the fetus has umbilical arteries, which is arising from the internal iliac artery and going to the placenta. You have umbilical vein, which is bringing oxygenated blood back from the placenta to the fetus. There is ductus venosus because it will bypass the fetal liver. We know that the blood doesn't have to reach the pulmonary circulation. The foramen ovale is a connection between the right and the left atrium because it has to bypass the pulmonary circuit. And ductus arteriosus connects pulmonary artery to aorta because most blood vessels, they bypass the pulmonary circuit in the fetus. In the 2nd month, the cartilaginous skeleton is ready. It is beginning to ossify into a hardened bone. And that is why you can see that by the end of 12 weeks of gestation, you can actually see the fetus. 3rd month is the month for fetal kidneys to be functional and breathing movements are coordinated. And my last of this topic is gonadal differentiation. I think Dr. Surveen would be talking in detail, but I'm only taking 3 slides for it. The gonadal stage is the period during which indifferent gonads will develop either into ovaries or testes. The phenotypic stage is induced in response to the gonadal differentiation. So the first thing to have to determine the sex of a fetus is the gene, is the genetic makeup. Due to this genetic makeup is the gonadal signals, and from the gonadal signals is the development of the phenotype also, along with the internal genitalia plus the external factors. So just we have to remember that in the formation of a male or a female fetus, it is always the presence of Y chromosome which determines testis. Please remember, if Y chromosome is absent or inactive, by default, the gonad will always be an ovary or a fibrotic tissue. So it is in the short arm of the Y chromosome is a sex determining region. It is the SRY gene. If this gene is present, it will activate the fetus, the fetal gonad. So till 7th week of gestation, the fetus is undifferentiated. You cannot make out that it will be destined to form an ovary or a testis. It is only from 7th week that the SRY gene on the short arm gets activated. It activates the testicular determining factor, converting into the male gonad. Alongside, there would be two ducts which will form in the fetus. Wolffian duct, please remember, will get activated if it is Y chromosome and will form male. And the Mullerian ducts, because males have a lot of anti-Mullerian hormone secreted from the Sertoli cells, so all the Mullerian ducts will be regressed and they will only be remnants. So if Mullerian ducts are present, it will form a female internal genitalia. If Wolffian ducts are present, it will form male internal genitalia. So just a recap, that fertilization, then there is SRY gene. If there is SRY gene, the undifferentiated gonad becomes testis. Otherwise, it would be an ovary. Testis secretes testosterone and AMH. Because there is anti-Mullerian hormone and there is testosterone, there is male genitalia, you feel like a male. If there is lack of testosterone and AMH, it is female genitalia, you feel like a female. And then you have chromosomal sex, gonadal sex, phenotypic sex, and the gender identity. What you do for yourself is what determines the actual identity of a person. So the keywords in this presentation, and then I will have a little bit of presentation on early pregnancy. You must know what is trophoblastic differentiation, what is primitive streak and its importance, what is gastrulation and importance, what is neurulation, what is metamorphosis, gonadal differentiation, and finally is the organ development. Now, few important points about early pregnancy. The pregnancy is established by the secretion of human chorionic gonadotropin, which is a placental hormone secreted by the syncytial trophoblast during 2nd week of conception. It supports the corpus luteum, which in turn supports the endometrium, maintaining the pregnancy. Now, if we look at maternal blood levels, so around 0 to 1 week of gestation, the levels can range from 0 to 50. The highest level of HCG is around 8 to 10 weeks. Why it is important? When you come across late ovarian hyperstimulation syndrome, you would see that maximum symptoms of OHSS happen by around 8 to 10 weeks, and then it starts to subside because the plateau of HCG has happened, and it is this plateau which stabilizes the symptoms of OHSS. Maximum level would reach around 2 to 3 months of pregnancy, and then they will plateau. So a value less than 5 is definitely negative, but in IVF we would say a value less than 25 MIU will hardly we will call that as a positive pregnancy test if it is done at the right stage after the embryo transfer. So in normal pregnancy, an HCG can be detected as early as 7 to 10 days. The levels, they rise rapidly. They normally exceed 100 by the time the first menstrual period is missed. It peaks to around 1 lakh to 2 lakh by around 10 to 12 weeks of pregnancy. Most of the times you can detect HCG in the urine with a sensitivity of over 25 milliIU per ml. The studies recommend that at what gestation can you visualize a gestational sac? There is no point getting repeated beta HCG levels just to see continuation of a pregnancy. By around 5 weeks, you can detect an intrauterine GSAC, definitely by 5.5 weeks, and cardiac activity you will be able to see by a transvaginal ultrasound by 6 weeks. A GSAC is usually seen with a beta HCG of about 1500 to 2400 by a transvaginal ultrasound, and an HCG level of above 3000 you can also detect with the help of transabdominal ultrasound. So why it is important? Because if you are not able to detect an intrauterine sac at this beta HCG, then one thing which you need to have in mind is ectopic pregnancy, and that is very, very important. And if you are doing multiple embryo transfers, please remember that you have to be very aware that there can be heterotrophic pregnancies with higher beta HCG levels. So the ultrasonologist should have this entity in mind when they are dealing with ART pregnancies. This is just a picture showing a GSAC at around 5 weeks, and it's a good intrauterine pregnancy. So now just last 5 slides about, where we are saying this is an iceberg, where I'm saying only 30% are the live birth. You will have most of the preclinical pregnancies. 30% would be lost even at the time of before implantation. Another 30% are lost after implantation before the first, beta HCG. There is about 10% patients who would have clinical miscarriages, and there is a 30% live birth rate when a patient, starts attempting pregnancy. So early pregnancy loss occurs during peri-implantation period before the pregnancy is clinically confirmed, and it is pretty common in humans. The risk of spontaneous abortion increases with implantation beyond this window. Why does this early pregnancy loss happens? Because there could be a lower expression of endometrial protein MUC1, suggesting a compromised endometrial epithelial function. The second could be that there is an epithelial impairment of barrier function, which could lead to a situation in which a poor quality embryo, which was not destined to implant, but will implant and will have a miscarriage. Number 3 could be that normal pregnant uterine decidua, which contains a lot of natural killer cells and large granular lymphocytes. So in patients with miscarriages, these could be increased disproportionately, but these are all in the research setting. So I don't think that in clinical settings we should go about looking for them or trying to find them. And this is what happens, that when we are looking at immunological causes of very early pregnancy loss, I'm saying, I'm not talking about clinical pregnancy loss. So that could be a lack of mobilization of immune cells, because please understand that pregnancy is an immunomodulatory situation. For mother, fetus is a foreign antigen. The mother really has to dampen her immunological status so as to accept this fetus. And that is why in mother, the TH cells, TH1 cells, they reduce, the TH2 cells increase, and there is an alteration in the natural killer cell population so that it does not reject the fetus every time. So there could be insufficient endometrial vascularization and insufficient adhesion responses, which are the interleukins. And the last is hyperactivation of uterine natural killer cells, and the embryo is destroyed because it is considered as non-self. So I think I've come to my end of my presentation. I hope that I've been able to put my point. I'm very open to discussion, and thank you very much for patient hearing. Thank you, Dr. Kuldeep Jain, Dr. Jayant Mehta, and Dr. Arne for giving this opportunity for me to discuss this topic today.
Thank you,
Thank you very much, Dr. Sweta, for a beautiful lecture. Sorry, Anya interrupted. Please carry on.
No, I just want to say it was a very nice lecture.
Thank you very much.
Excellent.
Thank you.
And I hope everybody has enjoyed. We are now moving actually from pure science to pure art. And I request Dr. Arne to introduce next speaker.
Yes, Dr. Survin is also a familiar face. The director and head of the IVF and Reproductive Medicine Center in the Max Multispeciality Hospitals in Delhi, Gurgaon. She's professor and School of Health Science in Ansal University. She has written 8 books, which is amazing, 75 publications. She's a program director for fellowships in reproductive medicine at the Max Hospital. She's a professor School of Health Sciences, as I said previously, the Ansal University. She's assistant editor in Fertility and Science and Research Journal. So there's a lot of peer reviews. She's the past editor of Tenswar and web editor, Indian Fertility Society. So she's got numerous awards. It's, maybe take too long to go through everything, but I'm looking forward to your lecture, taking us to the female fertility and especially the things of organogenesis, the ovary and the oogenesis. Please.
Thank you, Dr. Anne, for a kind introduction. And it's always a pleasure interacting with you, Jayant, Dr. Kuldeep, on this course. So I would be speaking on the female reproduction, basically the how the female reproductive tract works, what is the anatomy, what is the physiology. So this lecture would cover development of the female reproductive tract and formation of the basal gonadal architecture. Oogenesis and folliculogenesis, how does it occur? Anatomy of the female reproductive tract, regulating factors of female reproduction, that is the physiology of the female reproductive tract, how the hormones control the oogenesis. Now, function of the female reproductive system, the basic functions. So the reproductive system produces estrogen, progesterone, which is necessary for sexual development at puberty and reproduction later on. It stores and releases reproductive cells, which are called the oocyte. It allows fertilization to take place, and once that happens, it implants, nourishes, and protects the developing child or the fetus. So let's have a look at the development of the reproductive systems. The gonads arise from the intermediate mesoderm within the urogenital system of the embryo. The genital ducts arise from a paired mesonephric and paramesonephric duct, as you can see here. The mesonephric duct gives rise to the male genital duct, and the paramesonephric gives rise to the female. So a little bit about, I think Pankaj would be speaking more on detail, but since I was taking the reproductive tract, I thought I would just touch on how it starts developing. The first appearance of the gonad is essentially the same in two sexes, and in the initial weeks you cannot make out whether this is going to be a male or a female, and it just consists of thickening of this mesothelial cell of the peritoneum, which starts forming a distinct projection out here. This projection is known as the gonadal ridge, and this gives rise either to the ovary or the testes, as we'll see later. About the 7th week, it differentiates into either an ovary or a testes. The gonadal ridge starts getting pinched off from the mesonephris. However, some cells of mesonephric origin do join the gonadal ridge, as you can see, the yellow cells. So how does the ovarian follicle, since we are going to be talking about the follicle, how does it actually originate in the woman's body? During the early embryonic development, cells from the dorsal endoderm of the yolk sac migrate along the hind duct. These primordial germ cells, these yellow cells here, they multiply by mitosis because we want to increase the number of oocytes, and once they have reached the gonadal ridge, they are called oogonia. Now, these are the diploid stem cells of the ovary, and they give rise to the oocytes. They become surrounded by a connective tissue, the pregranulosa, in an irregular manner, and in this way they form the rudiments of the ovarian follicle. So this is the origin of the ovarian follicle, about which we are going to discuss later. So the development of the ovary commences at the mesonephris, right here, at the surface epithelium, where you see these yellow cells in the location of the future gonadal ridge. What we saw here, this is the gonadal ridge, is what we are seeing here. Okay, and this starts elevating. These GREL cells, they start proliferating, and the basal lamina underlying the mesonephric surface epithelium breaks down. So this basal lamina starts breaking down, and it allows the stromal cells, that is these green colored cells, to start penetrating the gonadal ridge. If you see, this is how it is forming. So this is the original lamina, this is the protrusion of the lamina, and then the breakdown. The GREL cells continue to proliferate, and primordial germ cells, PGC, these are the gray cells which you are seeing, these ones, okay, these ones. These start coming in between. Once they come in between, the mesonephric stroma, including the vasculature, continues to penetrate. So what happens is it goes around these cells and it forms these columns, and it penetrates towards the ovarian surface, and it encloses the oogonia, which are the gray colored cells, and it starts forming cords. Oogonia proliferate and the stroma penetrates further towards the ovarian surface, enclosing the oogonia and the GREL cells into ovigerous cords. The cords are surrounded by a basal lamina at their interface with stroma, but are open to the ovarian surface. Stromal areas, including those between the ovigerous cords, contain capillaries. So these cords, this forms the cortex, this part of the ovary, that is the superficial part of the ovary, and the medulla is formed by the middle part, which has the connective tissue and the blood vessels. So as we go along, we see that in the final stages, these cords are formed and become mostly single layered. So gradually they enclose only one cell within them, and the tunica albuginea, densely packed with fibers, starts surrounding them. Okay, the connective tissue starts surrounding them, and they get covered by a single layer of granulosa cells. So what is the timeline of the primordial follicle formation? Now the primordial germ cells colonize the somatic gonads at 5 weeks. Now at 5 weeks, this is what starts happening. The germ cells, they start forming a cyst. These cells start undergoing mitotic division because we need a number of germ cells in the woman, and they form cysts. And then once these cysts are formed, the meiotic part of the thing stops and the meiosis 1 starts. And this occurs between 10 to 12 weeks. This is when meiosis starts. So mitosis stops here and meiosis starts here. Then the cysts break down via apoptosis with germ cells to form the primordial follicle. So gradually these cysts will start breaking down. They will get enclosed by a single layer of cells, and they form the primordial follicle. That is the birth of the follicle happening in the fetus. So if you have a look at it, this is the fetal ovary. This forms the germ cells at about 5 weeks. There is a cyst formation. The cyst breaks down, and by 16 weeks, we have the primordial follicle with the oocyte and the pregranulosa cells. Now, this remains like this till the baby is born, and further till she attains menarche. So at sexual maturation, what happens is that there is conversion of the follicular maturation. So from the primordial follicle, they become primary, secondary, antral, and ovulatory follicles. So we'll come to that. So this is the follicle development. Now let's see what it is. So the primordial follicle is what we had at birth, right? After 16 weeks till puberty, you have a primordial follicle. This turns into a primary follicle at puberty and a secondary follicle. So primordial follicle is one layer of squamous-like follicle cells surrounding. This secondary follicle becomes multiple layers. So primary follicle is 2 or more layers. The secondary follicle becomes multiple layers. And the secondary follicle also starts developing a fluid-filled space between the granulosa cells. And finally, when this space becomes big, and there are 3 types of granulosa cell which are defined, it starts bulging. It is known as the Graafian follicle. And once it is released, it becomes the corpus luteum. So now very interesting. If you see that till this stage of the secondary follicle, that is before the preantral follicle, there is a gonadotrophin independent stage. And no hormone can act to do anything to this stage. It is only when it enters the preantral part that it is gonadotropin responsive. And there is some response to gonadotrophins. Gonadotrophins is the FSH, LH which we give. And subsequently, it becomes totally dependent. This part of the development is totally dependent on whether FSH is there or it is not there. So if you look at the timeline, in humans, a primordial follicle takes about 150 days to develop into a pre-antral follicle, another 120 days to form an antral follicle. A number of antral follicles will then compete for 14-15 days to become one dominant follicle. That is how the whole system takes place. So it's over months. This is the primordial follicle, which is small. It is 40 micron. The primary one is 100, secondary is 200, and tertiary is 400, with the antrum and a space developing, and it becomes even larger. So let's have a look at the secondary follicle, because clinically this is what we are concerned with. What we see, what we monitor is actually the secondary follicle developing. The fluid space in the follicle is what we are measuring when we are doing an ultrasound. So what does it consist of? A follicle usually contains only one oocyte. It is surrounded by granulosa cells. It has a fluid-filled cavity here. And it has, beyond the granulosa cells, there are theca cells. So the granulosa cells are divided into the membrana granulosa. Now, membrana granulosa is that part which is left behind once ovulation takes place to form the corpus luteum. Then is the outside the membrana granulosa is the theca interna. Now these are cells which we later will talk about, which are LH sensitive, and the theca externa, which is basically connective tissue which supports this follicle. So besides the membrana granulosa, the granulosa cells are divided into the cumulus ophiuchus and the corona radiata. Now the cumulus ophiuchus is this particular pedicle which is holding this oocyte within this follicle. So when ovulation takes place, this part, this column separates and breaks out and releases the egg. And what you clinically see under the microscope is this part, that is the corona radiata, the granulosa cells which are surrounding the follicle to nourish the follicle. Now these are directly surrounding, and all these are released at ovulation, and that is the one which our needle picks up and you see under the microscope. And it provides physical protection to the oocyte. Now follicular fluid, the antrum is a fluid space. This antrum is what is the follicular fluid. At ovulation, this fluid is released and it carries the oocyte out and it aids entry into the uterine tube. Now coming on to the theca interna and theca externa. Now the ovarian follicular endocrine cells forming the inner layer of the theca follicle surrounding the developing follicle within the ovary. So this inner layer is actually an endocrine organ for the ovary because it secretes, it is responsive to LH, it is vascularized, and it synthesizes and secretes androgens, which are transported to the granulosa cell, and here they are converted by the aromatase to estrogen. So that is how the ovary secretes estrogen. Each follicle will be secreting a certain amount of estrogen, but that estrogen, the origin comes from the theca cells as testosterone, and in the granulosa cells they are converted into estrogen. The theca cells do not begin their hormonal function until puberty. The theca externa, the one which is completely outside, it is basically connective tissue, smooth muscles and collagen fibers, and it forms the support of this follicle. So the next thing which we have to cover is oogenesis. So this is a comparison between what happens to the female egg and the male egg. Now, there is a 2N stage. It is multiplying. It becomes 4N by mitosis. And then there is a meiotic arrest at M1. It goes into the first meiosis, and there is at prophase, there is an arrest. This occurs till there is ovulation. Okay, so that means till she reaches puberty and starts ovulating, it is in the prophase of M1. And henceforth it becomes the primary oocyte. It releases one polar body at ovulation, then becomes a secondary oocyte. Again, there is a meiotic division and it releases the second polar body and it becomes a single M, which is a mature oocyte. What happens in the male? Here, one oogonia forms one oocyte and two polar bodies. In the male, there is a spermatogonia, again mitosis, meiosis, meiosis, and we get 4 spermatocytes. So this is the difference between male and female. So let's have a closer look at these events and related to the follicular development, the meiotic events relating to the follicular development of the ovary. Now the oogonium or the stem cells, they undergo mitosis because we want more in number. And this mitosis occurs only in the very initial part, as we said, just in the 5 weeks, 6 weeks period, the mitosis occurs. Subsequent to that, they undergo meiosis. And the primary oocyte is arrested at prophase 1 when the child is born. So that is the primordial follicle. The primordial follicle will contain an oocyte which is arrested at prophase 1 of the first meiotic division. This is childhood. Ovary is inactive. It remains as it is, as it was at birth. After that, each month from puberty to menopause, what happens? One primary oocyte, it develops, it grows into a growing follicle, into a secondary follicle, and into a Graafian follicle. And once there is ovulation, there is release of the first polar body. Okay, so the meiosis gets completed here by one primary oocyte each month at ovulation. That is the first meiosis. The second meiosis, and this meiosis, the second meiosis starts in the oocyte and is arrested at metaphase 2. Once the sperm penetrates the egg, this meiosis gets completed only if the sperm penetrates, not otherwise. So the second polar body is released once the sperm penetrates. Otherwise, the second polar body is not released. Okay. So this is the ovarian cycle. The secondary oocyte plus the small polar body. The secondary oocyte enters meiosis 2, stops at metaphase 2 until fertilization. This is at birth, mitosis. I am repeating it again. The primary oocyte, which is diploid, remember that. RNA replication occurs before birth. Following that, there is meiosis 1, which is completed after puberty at ovulation, where first polar body is released. This first polar body may divide further or may not divide. The secondary oocyte, meiosis 2, is begun in the tertiary follicle and is completed only if fertilization occurs. Secondary oocyte ovulated in metaphase of meiosis 2. So when the secondary oocyte ovulates, it completes the meiosis 1 and goes into meiosis 2 and is arrested at metaphase of meiosis 2. If the sperm penetrates, this meiosis is completed and the second polar body is released and the ovum becomes a haploid. How are the numbers through the progress of life? Like the oogonia divide by mitosis for the next few months and some differentiate into primary oocytes. So this is the increase which occurs in the initial weeks. By the 5th month, there are about 7 million primary oocytes, but most of them will degenerate during the next 2 months. Remember that. So the peak occurs, the mitosis continues till about the 5th month and the peak occurs there. In the stimulated cycle, 34 to 38 hours after triggering ovulation, a typical mature pre-ovulatory COC. I put this here because this is what you are seeing. So these are your granulosa cells which you see around your oocyte. And it is known as the corona radiata. And the more expanded it is, the more mature and appropriate the oocyte is. So the immature oocytes could be the geminal vesicles and they have tightly packed corona cells with little cumulus. Intermediate could be the nuclear maturity of these oocytes is metaphase 1. The corona and cumulus cells are again tightly packed, but in the mature one, where they're in metaphase 2, they will appear like a sunburst of corona cells with expanded and abundant cumulus like this. Post-mature, these types of oocytes are mature ones which have a dark cumulus oocyte complex in which the corona cells have begun to degenerate. So generally when you see a cycle, if you've done a good stimulation, you'll get about 85% to M2s, 10% geminal vesicles, and 5% maybe M1s. So this is a geminal vesicle. You would know this better if you're practicing embryologist, metaphase 1 and metaphase 2. Now coming on to the female fertility versus female age. 2 human embryos gradually lose follicles both before and after puberty. Like the loss of follicles starts from the 5th month onwards, because till 5th month the mitosis is taking place. At birth, a woman is born with 10 lakh or 1 million follicles. This is optimum, and gradually as she grows older, the follicles start decreasing, and when they reach 1 lakh, that is 10,000, 100,000, the fertility starts decreasing. This starts around about 30 to 35 years of age, depending upon the woman. And when they reach 10,000, around 40 years of age, the fertility is nearly towards the lower end. And once they have reached 45 and above, the oocytes are about 2,000, 3,000, 1,000, and that is when menopause starts coming on. So always remember, a woman is born with a set amount of eggs. A man produces eggs every 3 months. So sperms are generated every 3 months. But a woman, whatever she has, she has. Once she starts losing it with age, then we cannot regenerate that ovary, not as yet. Let's hope we have a science which does regeneration, but not as yet. And hence it is important that whatever process of fertility is to occur, it should occur at an earlier age with her, vis-a-vis a later age. That is why we take, in our cycles, we take donor eggs, because the woman probably has reached 40 or a premature menopause where her eggs have gone down. So let's come to the anatomical structures. So basically the female reproductive tract consists of this. There are ovaries. There are 2 solid ovoid structures, about 2 centimeters by 1 centimeter, so 3 by 1.5 also. And these ovaries are the ones which secrete estradiol and progesterone. Now, progesterone is secreted by the corpus luteum after the ovulation occurs. Estradiol, I have explained this already before. So, what is the histological structure of the ovary? It's very interesting. What I showed you at the development. So, these are your primordial follicles. So, a fetal ovary, if you would see it, this ovary would be studded with primordial follicles here. But in this diagram, they're showing you how it is maturing and becoming a mature follicle, and then rupturing for ovulation, and then forming a corpus luteum. So what is happening is that this part is known as the cortex, and this is the active part for steroidogenesis. This central part is the medulla, which contains the connective tissue and the blood vessels. Next is the uterine tubes. Now the egg, once it's released, it's carried, the fluid is released and it is carried into the tube. This tube has 4 parts. The initial part is the isthmus. Then is the long part, which is the ampulla. Usually the ectopic pregnancy harbors here. The fertilization takes place here in this tube. The infundibulum, which is the dilated lateral end, and the fimbria, which are generally over the ovary to catch the egg. So the fimbria sweep the ovary on the tube like this, and the fluid is released and the egg passes along and reaches the ampulla, and fertilization takes place within 24 hours after ovulation. The fertilized oocyte or the zygote reaches the uterus about 7 days after ovulation. So this is the histology of the fallopian tube, where you see the cilia. The cilia help in pushing or making a flow, establishing a flow so that the egg can pass through. Then comes the uterus. So the tube connects the ovary to the uterus, and the uterus is the place where the baby harbors, grows, and delivers. So it's a very thick muscular organ, and it has the capacity to grow the muscle hypertrophies, and it accommodates a 9-month pregnancy. So the part of the uterus, which is the major part, is the body, and this is the place where the implantation and the growth of the baby takes place. The fundus. The fundus is the rounded superior region. So when we are doing an embryo transfer, we generally leave the embryo here. Superior region, just where the tubes are entering. The isthmus is the lower part, which is a narrow part, this part. Now, the endometrium, that is the lining of the uterus, it consists of simple columnar epithelium, stroma of connective tissue, and endometrial glands. Now, the two parts, the stratum functionalis and the stratum basalis, and I'll come to this in a little more detail later. Then the muscular layer, and then the visceral peritoneum. So let's have a look at this. This is the lumen of the uterus, okay? This is the cavity. So here you have simple columnar epithelium, you have stroma, you have these endometrial glands. So this part till here is the stratum functionalis. Okay, this is the superficial part of the endometrium. The stratum basalis, which is the lower end of the endometrium, also is part of this whole complex. But it is the stratum functionalis which is shed at the time of menstruation. So this whole part is shed, and from the stratum basalis, it regenerates and comes up again. The myometrium is the muscular part, and it usually consists of 3 layers. Endometrial glands, the stratum functionalis, then the stratum basalis, and the myometrium. Now the cervix. The cervix is the lower, you can say the neck of this uterus, okay? It is on both sides. One side there is an internal os, that is the opening into the uterus, and on the other side is the external os, that is the opening into the vagina. Now, to relate clinically, this external os is what you see when we are doing an embryo transfer, and this is the place where we put our ET catheter in, and from here we negotiate and reach here before we leave the embryo. The cervical glands also are responsible for secreting the mucus, which we are, clearing out and cleaning before we do the embryo transfer. The lower part, that is the vagina, and this is the part where in a natural intercourse the sperms are deposited. It is flexible, expandable, and once the delivery takes place, it expands to 10 centimeters. The upper ends are known as the fornices. This is the external genitalia. You have the prepuce, the clitoris, the vestibule, the urethral orifice, the hymen, the vaginal orifice. This is from where you enter for the ET, and the opening of the duct of the greater vestibular. I also wanted to tell you that this fornix is important because this is where our ultrasound probe goes. We put from here through the vagina for the aspiration. Now, coming on to the fourth part of the lecture, the physiology of the female reproduction after puberty. Each month, the hormones cause several follicles to develop, which triggers the primary oocyte to resume meiosis. So what happens is that a number of follicles, about 7, 8, 10 are recruited in each ovary, and they come up, but only one of them is selected to become the dominant follicle and ovulate. So what do we do in IVF? We give external hormones so that we can bring all of them up and enlarge all of them. I'll come to that later. When the cell divides, all cytoplasm and organelles, they stay with one of the new cells, and the other cell is just DNA. So what are we discarding in the polar body? We are discarding only the DNA. All the important things are in the oocyte. The secondary oocyte is the stage at which ovulation occurs. Now, let's see what is the ovarian cycle. Each month, the FSH, that is the hormone from the pituitary, it stimulates these primordial follicles to grow, and ovulation is the release of the egg, which occurs with LH. Again, LH is the hormone from pituitary, which causes the ovulation to occur. And once the ovulation, the remains of the follicle get transformed into actually an endocrine kind of a structure, corpus luteum. This corpus luteum is responsible for the progesterone to be released, and this is what causes the secretory changes in the endometrium so that there can be implantation of the embryo. And in our clinical medicine, we generally, when we do the ET, post that we put the patient on progesterone. That is to simulate the corpus luteum. If a pregnancy occurs, it produces progesterone to maintain the wall of the uterus during early period of development. So, corpus luteum will live only if the pregnancy occurs. If the pregnancy does not occur, what happens? It degenerates. And then it is known as the corpus albicans. That is the degenerating corpus luteum, and it just clumps into a scar tissue. So here again, they're showing this is the proliferative part of the phase where the first part of the cycle, when the follicle is growing, what is happening to the endometrium? The glands and blood vessels are scattered throughout the functional zone with little or no branching. Okay. But under the effect of progesterone, what happens? The new glands, they start forming and the endometrium starts getting thickened. These glands, they enlarge, they have branches, and they prepare the endometrium for the implantation. If no implantation occurs, then this upper part, the functionalis, breaks down and there is menstruation. Now, how is this whole process of ovulation, or developing the follicle, ovulation, who controls it? Basically, the hypothalamus and the pituitary. The hypothalamus acts like a computer. It analyzes signals, and it releases what is known as the GnRH, gonadotrophin releasing hormone. And it does this in a cyclical manner. In the initial part of the cycle, the FSH is increased, and through portal circulation, the pituitary hormones release FSH and LH once the GnRH is secreted. And this causes the cyclical changes in the ovary of estradiol and progesterone. So let's have a closer look at this. So the GnRH secretion is very short, 2 to 4 minute half-life. So it is continuously, fed in. But because it is continuously every 2 to minute, it is being fed in, it can be controlled by feedback mechanisms. So there are 3 main feedback mechanisms. The long loop, as you see from here, this long loop is the action of the target hormone on the hypothalamus and pituitary. Now, what is a target hormone? What is the target organ in this whole case? Is it the hypothalamus? Is it the pituitary? Or is it the ovary? It's the ovary, because this is the final organ which is being impacted, which secretes the estrogen and progesterone. Once estrogen and progesterone are released, they go back and they inhibit GnRH release, and that causes a decrease of FSH release, and that brings an end to the cycle. So the short feedback is this is the short feedback loop. The FSH and LH go and they suppress the hypothalamus and they prevent release. And then there is a very ultra short feedback where GnRH itself only starts suppressing its release. So it's an auto phenomena which takes place over a period of 15 to 30 days, where every 30 days this FSH is released and inhibited. So what is FSH? It is secreted from the interior pituitary, initiates the follicular growth through the granulosa cells, involved in selecting the most advanced sensitive follicle to proceed for ovulation. So the luteinizing hormone from the interior pituitary stimulates the theca interna to synthesize and secrete the androgens. So if you remember, initially I told you that the LH from the anterior pituitary goes, it acts on the theca cells. The theca cells, they release androgens. These are transported to the granulosa cells. And in the granulosa cells, the testosterone and the androstenedione are converted into estrogen. So let's have a look at the cycle here. Now this is a maturing follicle. What happens here? The FSH starts rising and it recruits one follicle. One follicle is recruited. This follicle starts secreting estrogen. Okay. Now, once estrogen is at a certain level, the LH increases and there is a LH surge. This LH surge causes ovulation. Once ovulation occurs, then what happens? The progesterone starts getting secreted by the corpus luteum. Okay, that is under the influence of LH. So, if you look at it from here, waning production of the estrogen and progesterone by the corpus luteum. So, what's happening here? Let's see. The corpus luteum, now there's no pregnancy, so it is becoming the corpus albicans. So it degenerates. That means all the estrogen, progesterone which was being secreted there by the redundant granulosa and theca cells has come down. Okay. Now once it comes down, what happens? Once it comes down, this feedback is removed. Once this feedback is removed, GnRH is released and then FSH is released. So what happens? There is a rise in FSH. This FSH starts developing follicles. This induces expression of FSH receptors on the follicles and it stimulates the follicular secretion of estradiol. Okay, so the granulosa cells, they start producing estradiol. The estradiol inhibits the secretion of FSH and LH. So what happens? Let's go back to this diagram. The estrogen starts increasing. So what is going to happen when it's here? The FSH starts coming down now because these are high here. So once the FSH starts coming down, one follicle evolves and a dominant follicle, it is destined for ovulation and the remaining undergo atresia. This matures and secretes increasing amount of estrogen which peak towards the end of the follicular phase of menstruation, and estrogen exerts a positive feedback on LH, generating a dramatic pre-ovulatory surge. So here estrogen does not do a negative feedback. Once it's reached this level, there is a surge of LH, and this surge of LH is what causes the ovulation to occur. So two concepts which you must understand in the body to understand how we stimulate and what impact the stimulation can happen. There are two things: FSH threshold and FSH window. Now, FSH threshold is the level of FSH which is below which no follicular growth can happen. FSH has to be above a certain level. Some have postulated that this level is 7.8, but we never measure it. Initially, the FSH values may be below, but then gradually, as FSH is secreted in the initial part of the cycle, as you can see here, it goes up, it reaches a threshold and it makes this start growing. Now, this is what happens here. Okay, it goes up, threshold, this follicle starts growing, the rest of them are not dominant, so they don't start growing. What is FSH window? FSH window is the number of days that the serum FSH levels are above the threshold, and it determines the number of follicles which are activated. So if I've increased the follicle in a natural cycle, FSH is increased, it will at some stage come down once the dominant follicle is recruited. The rest of the follicles will all die out. What am I doing in an IUI? In an IUI, I am giving either clomiphene or I am giving FSH injections to increase this window so that I get 2-3 follicles. What am I doing in IVF? I am giving FSH injections so that this window remains open right till the end so that I can recruit all the possible follicles present there. So you understand this is the correlation with the clinical medicine. So how does ovulation occur? Now, under the influence of LH, we know that LH is rising. There's that peak which happens. The primary oocyte enters the final stage of the first meiotic division and divides into the secondary oocyte and the first polar body. This is what happens. at time of ovulation. It induces also LH induces a release of proteolytic enzymes which degrade the surface of the follicle and stimulate angiogenesis, and the follicular wall opens up and ruptures. The oocyte adheres to the ovary, and muscular contractions of the fallopian tube bring the oocyte into the fallopian tube where it gets fertilized. Now the luteal phase, that is also very important. What happens? Post the LH surge, there is conversion of the granulosa cells from predominantly androgen converting cells to predominantly progesterone synthesizing cells. So if you remember, I had told you that there is an aromatase inhibitor, and once the LH acts on the theca cells, and the theca cells produce testosterone, which gets converted in the granulosa cells to estrogen. Now, this function of converting the androgen to estrogen stops after it has entered the luteal phase, and the granulosa cells start producing progesterone instead of estrogen. There is expression of new LH receptors which foster increased progesterone synthesis, and there is reduced affinity of granulosa cells for estrogen and FSH. So FSH stops acting. Increased progesterone secretion with some estrogen secretion. It peaks between 5 and 7 days post ovulation. There is high progesterone level. This exerts a negative feedback on the GnRH. That is the long feedback loop. As the GnRH pulse frequency decreases, FSH LH secretions decrease. Lacking stimulation by FSH and LH, after 14 days, the corpus luteum dies out, it gets atretic, and it becomes a corpus albicans. When the decline of both estrogen and progesterone levels is very, very important, because this is the negative feedback control on FSH, which is removed. And then the FSH, it's very important for the next cycle to occur, that this control is released. FSH starts acting, it starts recruiting more follicles for the next cycle, and we go on to the next cycle with the FSH increasing. So that part is very important. The degeneration of the corpus albicans is what causes the removal of the suppression of FSH. So the endometrium, in all this process, what is the endometrium doing? In the follicular phase, when the follicle is growing, there is proliferation of the endometrial glands, there is proliferation of the connective tissue, the lining starts becoming thick, the glands become elongated, and the spiral arteries also elongate. So if you notice, we are measuring the endometrial thickness on ultrasound, and we keep saying that, oh, it's 5, it's 6, it's 8, it's 9, because this is what is happening. It is increasing, and we can measure it on the ultrasound. In the luteal phase, what happens? Because it is predominantly progesterone, this progesterone induces the endometrial glands to secrete glycogen, mucus, and other substances. And because they are secreting so much, they become tortuous, they have large lumens. The spiral arteries extend into the superficial part of the endometrium. As the estrogen progesterone levels start to decrease, the endometrium starts to involute, and there is a breakdown of the endometrium and ischemia, and that causes the menstruation. In the menstrual phase, the spiral arteries rupture secondary to ischemia, and they release blood into the uterus. Just one slide I put here to show you the ultrasound findings. The early proliferative phase, you'll see that the endometrium is thin. It is not well defined into 3 layers. As the endometrium thickens, the glands thicken, this part increases, and we get an optimum 3-layered endometrium. Post ovulation, because of the secretory activity, the glands getting full, it gets echogenic. So these 3 lines are no longer well defined. So to summarize the functions of the female reproductive system, the ovary produces female gametes and sex hormones, estrogen and progesterone. The oviduct is the site of fertilization and the movement of the egg and embryo from the ovary to the uterus. The uterus is the site of implantation of the embryo, fetus development, and the muscular wall contracts to push the fetus out during birth. Cervix directs the sperm into the uterus from the vagina. The mucus plug during pregnancy prevents infections, the secretions from the cervix, and dilates at the beginning of birth to let the child out. The vagina is the site of deposition of the sperms during intercourse. It's the birth canal. It stretches during birth to allow passage of the fetus out of the body. This is a very nice diagram which depicts everything which is happening. So you can see that there is low E2 right in the beginning, and the follicle stimulating hormone, it increases, and it causes development of this follicle. At the same time, it causes the thickening of the endometrium, and the LH also will start increasing at the later half of the follicular phase to give an LH surge, because the estrogen is increasing. The estrogen causes an LH surge. This LH surge causes ovulation. Once ovulation has occurred, the corpus luteum is formed. This corpus luteum causes secretory activity, glandular activity in the uterus lining, and it prepares the lining for the baby. And the corpus luteum secretes estrogen as well as progesterone as support hormones. And once the pregnancy does not take place, the corpus luteum degenerates and becomes albicans. The estrogen and progesterone decrease, and because they decrease, the negative feedback on FSH, because FSH is very low here, it is removed and the new cycle is started. Thank you. So, I don't know if there are any questions.
Thank you very much, Dr. Survin. Anne, would you like to take?
Thank you for the lecture. And I tell you that I'm teaching the same thing for medical students. So you gave me quite a few tips for one next time. Thank you. I enjoyed it.
Thank you, Dr. Anne. Pleasure.
The questions would be answered after Dr. Pankaj's session. We are collecting everyone together. So if you don't mind waiting at that time, we'll have a Q&A at that stage. Thank you very much. Everyone has appreciated your talk. Much appreciated. Thank you. Dr. Kuldeep, if you could introduce our next speaker. Unmute yourself, sir. Dr. Kuldeep, if you can unmute yourself, thank you.
Yeah, thank you, Dr. Jayant and Dr. Survin for a wonderful lecture. So our last lecture for today is by Dr. Pankaj Talwar. He is a very well-known figure of Indian Fertility Society. He is past General Secretary of Indian Fertility Society, and now he is the Vice President Indian Fertility Society. Apart from that, he is conducting a lot of academic programs. He is on the editorial board of our journal also. He is member advisory committee, ICMR, member infertility committee, FOXY. At present, he served army for very long years, and now he is medical head at C.K. Birla Institute and IVF Center. Dr. Pankaj, are you there? Hello. Has Dr. Pankaj joined? Rajesh?
Not yet, sir. We're trying to call him.
Yeah, please call him. He was supposed to join. Okay, so Dr. Jayant, I think it may take little time, so we can take up a few questions if there are any from Surveen and Sweta if she is there.
Yes.
Yeah, I'm
Anne,
there, sir.
would you like to? Anne, would you like to come in? Dr. Anne?
I'm here.
Would you like to, you're here, good. Would you like to start the Q&A for all of us?
Do you mean by reading the questions that have been asked, or?
Yes, yes, and also for a general discussion.
Well, let's open up for the audience. Please, you can now start to ask questions. Now you have the chance. So, Jayant, have you collected the questions?
I have one question here. If I can initiate it. This is from Dr. Rekha Maratha asking that does the small FSH peak with the LH spike at ovulation responsible for fresh recruitment of follicles for the next cycle?
So I think I'll take that one because it's
Sure.
covered by my lecture. So no, the FSH peak is not, the one at ovulation is not responsible for recruiting new follicles because that's too early to recruit. It is the FSH which starts rising towards the end of the cycle in the last 5-6 days, when the estrogen and progesterone come down because there is no pregnancy and there is degeneration of the corpus luteum. The corpus luteum is what is producing the estrogen and progesterone. So once the estrogen and progesterone come down, the negative feedback in the hypothalamus and the pituitary by the estrogen and progesterone is removed. Once that is removed, the GnRH is released from the hypothalamus. That causes the increase of FSH towards the end of the cycle, and that causes the recruitment. So recruitment takes place in the previous cycle, but just a few days before it. It's not in the middle of the cycle when ovulation is taking place. That FSH peak is there because of the LH peak. Both of them peak together.
Okay. Again, from the same candidate, we have a question to Dr. Keshav. If I have only 2 embryos, one good quality and other poor quality one, should I only transfer just one good quality one, or won't just one embryo transfer not reduce my success rate?
Thank you, Dr. Rekha, for that question. Now, this is a very tricky situation because you only have 2 embryos. Now, like I mentioned in the previous presentation, when we are thinking of endometrial selectivity, this might cause like the endometrium to stop accepting the embryos, and that's something that's still a developing concept. So if I talk about just practical experience, I would transfer 2 embryos. It also depends on the stage of the embryo. If you have 2 blastocysts and there's one blastocyst which is poor quality, which is like a CC blastocyst, then I would probably avoid and I'll only transfer a good quality blastocyst. But if it's like a day 3 embryo or even like a day 2 embryo, then I might still choose, looking at our Indian setup, I might choose to transfer both together.
Well, there's epidemiological evidence that if you transfer one good and one bad, it has a negative,
yes,
not only on the implantation and pregnancy, but also on the outcome of the pregnancy. So a little premature births, a little premature babies. So I think if where I'm living, we would definitely go just for one, for many reasons.
I think in my center, if it's a cleavage stage, one poor and one good, we would either go to blastocyst and look at the growth of the blastocyst and transfer one. Or if we really have to do, we transfer one good cleavage stage embryo and we follow the poor one. If it forms a blast, you freeze it. If it doesn't form a blast, you discard it.
So it all depends on the patient profile,
yeah,
and also the setup and the financial aspects. So for a country like India, all of these factors will determine. But for a Western country, insurance backed, etc., I would only choose to transfer one based on the evidence that I presented in the lecture.
Dr. Kuldeep, any comments?
No, I think as Keshav has said, depending on the situation where you are practicing and what is the growth of the developing embryo. But evidence suggests that if you are transferring a good one, only good one, that is enough to give a pregnancy. So you should not transfer the slow growing embryo or a bad quality embryo. That might interfere with the implantation process.
We have a question from Shiva. Pyruvate in high quantity in oocytes. Day 1 and day 2 lactate will be more in day 3, 4 or 5. Question to Keshav.
Okay, Shiva, thank you for that question. I think you're misunderstanding it a bit. It's in the culture media that pyruvate has to be present for the cleavage, like initial cleavage stages. So from day 0 to about day 2 or even the start of day 3, the embryo is going to actually take pyruvate as the prime source of energy. From day 1 also it can start metabolizing lactate into pyruvate and use it for energy. So that also can be done. But from day 3 onwards, the prime source of energy is actually going to be glucose. So that's how you have to plan your medias. So if you're using a sequential media, that's the kind of energy sources that changes through the sequential media. But in a single step media, you have everything which is present together and the embryo just takes in what it wants.
A further question from Dr. Rekha. Anyone can answer this. If I do a day 5 transfer on the 15th of a month, on the 30th, the beta HCG is 15. On the 6th of the next month, it is 95.5. How should I interpret the results when TVS on 6th is showing a very small, just a black dot?
I can just attempt that. So I don't remember the dates. So if it is day 11 of a blastocyst transfer and beta HCG is 25, then practically this pregnancy will not go in. But the only thing I have in mind, I would see a doubling after 48 hours. If it has spontaneously fallen down, then I will discontinue all the progesterone support. If it is showing doubling or less than doubling, then I also need to keep in mind ectopic pregnancy, because once in a while we do come across that. But at the completion of 5 weeks, that is 1 week from the actual date of beta HCG, if a very small dot is seen, I think correlation with beta HCG, either it is pregnancy of unknown location or it's a failing pregnancy. And this is how I would take. So TVS, 5 weeks, they should see a sac.
This is actually day 15 of the blastocyst transfer, where beta HCG is only 15.
Which is like almost negative, yeah.
Negative, negative of biochemical. And day 21, it comes to 95.
Yeah, but I think you need to follow these beta HCG level till there is a declining trend, because the low beta HCG and less than doubling beta HCG in 48 hours, they need to be followed to exclude ectopic pregnancy, because that happens once in a while. And if you don't follow these pregnancies, these beta HCG level, then your patient may end up in a ectopic pregnancy and in a poor situation, poor clinical situation.
Dr. Surin, how would you interpret that?
So it's definitely a low beta HCG. It's not a negative beta HCG. We cannot ignore it. The doubling time is not in sync. It is it should have doubled more. At day 21, expect a higher beta HCG. So I would look at it as either an ectopic or a failing pregnancy, and I would follow it through with another beta HCG probably to see how this beta HCG is behaving. I would continue to follow it till the beta HCG has dropped down. I would not leave this patient.
Anna, would you like to add something to that?
Well, no, not that one, but there's another question here from Silly Didman. Should we still prefer day 3 freezing and day 3 embryos transfer, taking embryo genome activation into consideration, or prefer day 4 and 5 for better selection, even in the cases where embryos are less in numbers? That's a classical question. Mm-hmm. Day 2, 3, 4, 5. Anyone care to comment on that?
I personally would prefer to follow an embryo till blastocyst and freeze the blastocyst. The survival rates are fantastic with blastocyst cryopreservation, and it also tells you a little bit about the potential of the embryo. Now, if the embryo is forming a blastocyst, obviously it has a higher potential as compared to a day 3 embryo. So that would be my take on it. I would not consider, now I know that in India people do freeze day 2 embryos as well. So if I want to grade it, my preference is a blastocyst, then a day 3, and then like a day 2 embryo. It also depends on the number of embryos that the patient has, how comfortable the clinicians are with like following the embryo till a blastocyst. Now there are a lot of determining factors for a country like ours. But personal preference is a blastocyst.
I agree.
Yeah, so in my center, we don't do day 2 freezing at all. So I think if she's a young patient, like say 30, below 35, and she has two good, decent day 3 embryos, then we do grow them to blast day 5 or day 6. If suppose she's a 38-year-old lady, she has one decent, one average embryo, and she's not even going for PGTA, then the embryologist just might tell us to do a fresh day 3 transfer. But if we are going to day 5 in that case, then we have noticed that day 5, they slightly bring lesser pregnancy in fresh transfer. So we would like to freeze that. So I think cleavage stage, day 3, fresh or frozen, fresh, a bit comparable pregnancy rate to blastocyst. But if we go to blastocyst, we like frozen. This is how we do. And depending on the age and whether it's her first IVF cycle or are we dealing with RIF? In RIF, we 100% want to go to blastocyst, no blast, no transfer.
Yeah, we generally go to blastocyst. We don't do a day 2.
Yeah, Keshav, have you compared the day three thaw taken to blastocyst and then transfer, or blastocyst transfer?
Sir, thank you for that question. Now, if I talk about 2 years or 3 years down the line, that's when we were still assessing day 3 versus still freezing day 3 and following it to blastocyst and then transferring blastocyst. But now we've stopped doing that. Most of the cases, it's just followed to blastocyst. We freeze the blast and then transfer blastocyst in a FET cycle. So my data from 2016 or 2017, yes, definitely suggests that blastocyst results are better. Even when I'm freezing day 3 embryos, most of the biggest challenge that I faced was that I was probably thawing about 4 or maybe even 5 day 3 embryos and culturing them to blastocyst. And then I always had extra blastocyst, which was supposed to be refrozen, which I didn't want for my patients. So my piece of advice is that it's best to follow them all to blastocyst, freeze the blast and then transfer that.
Dr. Pankaj has joined us. So, Dr. Pankaj, could you care to comment on this question, that the discussion that we're having? Dr. Pankaj?
In our center, we don't do long culture. The only situation where that is happening is if the patient has already had one child and for some reason 2 day 3 embryos were frozen and she's come for second conception, then we culture a long culture to blastocyst and we transfer 1, because we do 70% single embryo transfer in our center. So that is the only situation. But if it's a day 3 frozen, then it's transferred on day 3.
Yeah, I don't see the significance of actually freezing day 3 and then following it up till blastocyst. Why not just follow it initially to blastocyst and freeze the blast? That's my take on it.
I think a stricter grading criteria on the day of freezing is much more helpful, which will reduce the number of embryo frozen but will increase the implantation rate.
And that ultimately helps the patient out.
Yeah.
There is a question from a candidate, Sesha. She's asking, I know whether primordial germ cells origin from epiblast and then reaches yolk sac, or is it directly from the yolk sac? Few books are giving yolk sac and few are giving epiblast. What to follow? I guess the question is for.
So I also looked up, and what I found was that they are from the extra embryonic, they are the cluster of extra embryonic cells, and they are totipotent. So they are somatic stem cells which are present in the epiblast, and then they are seen on the site of the definitive secondary yolk sac, possibly migrating through the mesoderm and endoderm. So right now I would call it that they are epiblastic cells, which are basically stem cells. Somatic stem cells.
There is another question for you, Sweta.
Yes,
first
sir.
polar body is released at ovulation and second at fertilization. Usually one is seen at first, one degenerates, but yes, as far as I know, you can very well see the second one too.
Yeah, I that is what I had written the answer, but I think Keshav can also say, because you will see, because when the second polar body will be released, even the first polar body will divide, but because it is degenerated, we don't see it. Otherwise, seeing 2 polar bodies, there is no harm.
Okay, so this question basically from my interpretation of this question is right after denudation, if the embryologist is seeing an oocyte with two polar bodies.
Okay.
If that is the question, it probably signifies parthenogenesis as well. And I would be skeptical of injecting such an oocyte with a sperm, because it could be an error of oogenesis. So even if you inject such eggs, I would really want you to keep them separate and follow the embryo, and maybe like deprioritize the embryo that's formed for transfer from such a situation. There are situations where I think there's another question similar to this, where you have multiple polar bodies within the oocyte, and those are generally aneuploidic, and they are generally discarded, and they're not used for ICSI.
So do we interpret it this way, that if there's too much of polar body, that means the too much of genetic material or cytoplasm has gone into the polar body, compromising the competence of the oocyte? Is that how you interpret?
Also, if parthenogenesis has taken place, then obviously it's divided, no? So it's not what it was.
But that would look like a two-cell embryo rather than two polar bodies.
Would be the start of
Yeah.
parthenogenesis, because this is right at like 38 hours post HCG. And if you follow such embryos, even if you don't touch them, they might divide into a 2-cell, maybe up to a 4-cell, and then just arrest at that point. So you're never really sure whether these embryos are competent or not. The second part that you were asking was whether this is extra genetic material. Yes, this could be like, these could be aneuploid oocytes where extra genetic components have been extruded in the form of polar bodies. But that generally tells me that this egg is not as competent as it should be, and I would not really choose it for ICSI.
Okay.
I think
Doctor.
Pankaj has joined. Pankaj, are you okay?
All okay, fine, sir. Hi,
Yeah.
Dr. Arne. How's everybody? I'm all right, sir. Hi, Keshav. Hi, Surin.
Hello, sir.
Hi, long time, everyone. Hi.
Hi.
I will chat, sir. The moment you permit me,
I welcome you. I welcome you to this program, and Dr. Pankaj has been associated with this program since its inception, and you have been already introduced to the audience, so you can just go ahead.
Thank you so much, sir. This is my CV. Am I seen by all?
Yes.
Am I audible? Okay, thank you so much. So the topic which has been given to me is a pretty complex one, and being a clinician, I'll try to take the topic towards the clinical implications of all these things. I've been told to speak on section 3 of the Eshre curriculum. I have to speak on embryonic stem cells, fetal ovary, fetal testes, and gamete interaction, and until first cleavage. What I heard was that a lot of things have already been covered by Dr. Shweta initially. So that will make my job much more easier. So I'll be covering the following topics, as you can see on the screen. Looking at the importance for the Eshre exam, and as well as our clinical understanding of the issue, I'll be covering the topics in the following order: 3 and 4. So thank you so much, sir, for giving me this opportunity to speak here. And it's wonderful seeing everyone here. And for all the delegates who are present, very good to see that we have crossed the figure of 100 this time. And when we started, we were always having 10, 15, 20. This speaks volumes about Dr. Kuldeep's, Dr. Arne's, and Dr. Jayan's initiative. So without much of ado, I'll hit my topic. The topic given to me is, the first topic will be the introduction to the embryonic stem cells. Embryonic stem cells in the past 15 years have captured our mind, captured our imagination, because they are immortal. And we can do anything with them. It's something like we can keep culturing them, keep using them, keep subculturing them, and then taking them towards any direction. We can make a fetal bone, we can make liver, pancreas, kidney, retina, an eye lens, anything from a pluripotent stem cells. And human embryonic stem cells are actually pluripotent stem cells. This is what we have to know. So how does it work? When you look at this picture, which I took it from the website of NIH, on the day 0, we have an oocyte which gets fertilized by an sperm. It becomes day 3, day 4, or day 5 embryo. When I was working on embryonic stem cell lines initially, we were working on totipotent stem cells. That was year 2002 and 3. But later on we shifted, by 2008, we shifted entirely to work on inner cell masses. So what we do is, as Keshav covered beautifully, we grow a cell to a blastocyst. We cut open the covering, the shell of zona pellucida by either trypsinization, or we use something called pronase. That's the way we were using pronase initially when we started. So by using trypsin or pronase, the whole thing is dissolved and we're left with an inner cell mass. Now, nowadays, we are using laser. We can cut it open and the ICM is extracted. Or still, what we people do, we can do is that we can take a plate in which we'll place the blastocyst directly after puncturing it. And they say that it can, ICM will go and get adhered to the matrigel or the feeder layer and starts growing. So the most beautiful part about these stem cells are, human embryonic stem cells, are that they are derived from the blast. Though we have a technique called reversal of potency, where we can take a skin cell, and reverse its growth, and from a totipotent cell, from a skin, we can make a pluripotent stem cells. This is called induced pluripotency. This is a Japanese technique and routinely people are using it nowadays. So any cell can be taken back into the pluripotency and converted into a sort of an embryonic stem cell mass which is pluripotent. These cells have got an advantage that they have their origin from the blast, from the ICM. Bit difficult in the humans, but in the mouse model, when we work on the mice, it was very easy to handle them. They grow very rapidly. When we passage them, they are very faithful lines. Human lines are a bit tricky. They move left and right. They have the capacity to self-renew. One cell will multiply into a similar cell, and we normally take them to around 20 plus passagings. We call it passagings or passagings. We keep growing them, and when we are sure that they are not changing, they are not maturing, and they are maintaining their qualities of pluripotency, then the stem cell line is declared open for circulation in the world. These are bottled, and we normally bottle them into these vials like this, cryo vials, and they are sold all over the world for $40 to $70. Till now, we have got 100 plus lines in the world which are floating around. Majority they come from Israel and USA and Canada. You can order them online in case you want to order them. They are delivered to you by Blue Dart. Excuse me. Pluripotency is a quality in which a cell can divide, can multiply, and grow into any type of cell line which you desire. It can grow into an ectoderm, mesoderm, or endoderm, and make the appropriate organs. We know what ectoderm can make, we know what mesoderm can make, and we know what endoderm can endoderm can make. So what we do is, we take a cell, group of cells, we multiply them. We multiply them into a state where they can take any direction. And when we are ready, we can induce them by giving them some igniting factor. We ignite them. Suppose we add BMP, that is bone morphogenetic factor, or we add some other factor depending upon what we want to make. They'll start growing into that particular stem cell line. Our favorite was making cardiomyocytes, because cardiomyocytes are easiest to make, followed by the neurons. And third easy thing which we could make was always the bone. So any pluripotent stem cell mass, in case you leave it on its own, it will make a cardiomyocytes, and they are beating cardiomyocytes. The colony will grow and will find it beating like this. And it beats at the human heart rate. They are so sensitive, the cardiomyocytes, that in case you keep it in cold surface, they go into bradycardia. And in case you culture them properly, they have a good heartbeat, the whole colony is beating. The advantage is that when we have pluripotent stem cells, we can do a lot of research. We can put some drugs into them. We can look at DNA repair mechanisms. We can make stem cell lines of particular lineages which are defective. Suppose we get a lineage from somebody who's got a Klinefelter syndrome or a Turner syndrome or a Down syndrome. We knock the DNA. We know that this stem cell line has got a problem. We knock that DNA and repair it. This is what is being done in Israel at Haifa, from where I train. And this is a very good technology where we can repair these stem cell lines, pluripotent stem cell lines, and we see whether the Down syndrome genes, they disappear or not. The Klinefelter goes or not. So the genes can be cut and reformatted. Excuse me, I got a bad cough. Please ignore me for that. So what do we do here? In very simplistic terms, English is always there. In very simplistic terms, I like you to take you to a journey of how to generate stem cell line, embryonic stem cell line. So what we do here is that we have grown an ICM. So we cut this ICM open either by pronase or by laser. These cells are removed. Like you do PGS, you take some amount of these and these cells. Here we take these cells only. We take this cluster out of ICM. They may contain around 100 cells. And we plant them over an 100 mm plate, the Falcon plate which we use. Falcon, you can use blue or green line, both are okay with this. We coat this plate with something called matrigel. The place where I learned and worked is Haifa, and they are the ones who discovered human foreskin fibroblast layer. Foreskin fibroblast layer they made, Dr. Amit Mikhal. In case you go to the websites, you'll find she is a very big person. She is the initiator of the stem cell lines in the world. So we coat either mouse embryonic feeder cells or by the matrigel or human fibroblast layers, and we put this thing on this. So this colony will start growing. It's the ICM colony will start growing, and we seed every day. And after around 4 to 5 days, we find that this colony is going into senescence, is becoming old. It's becoming mature now. So when it becomes mature, it changes color. So that's a very fine time. It's a look which we have. We see it on an inverted microscope every second day. The moment it becomes mature, we cut out this colony with a blade, or we use trypsinization. We use trypsin, and this colony is removed. And this colony is then put in a tube, and we put DMEF media is called DMEF media is put into this, and colony is broken down like we resuspend a pellet. And then we see it on a cytometer and see how many cells are forming in this. We see on the grid, do the cell count, and then we replate them on a same plate like this. We take few cells from here and look for certain genes of pluripotency. What are the genes of pluripotency? They are octamer 4, nanog. They are the ones which nanog and octamer 4 are the two genes which we really carry out, and we look at whether they are expressing these genes or not. In case they don't express octamer 4 or nanog, we know that this has escaped and this is no longer pluripotent. Though there are multiple other genes are also available, we take out the DNA and check it. We take out the RNA and check it. We do this process a large number of times over a period of 1 or 2 months. And when that line is able to sustain the stability in terms of expression of octamer 4 and nanog, and the karyotyping is the original karyotyping, we are sure that we are dealing with a pure stem cell line which is pluripotent. That's the time when we declare it open, and then it can be sold all over and used for research. So with this, I'll go to some theory about embryonic stem cell lines. So they are grown for a couple of months. Every week we're doing a passage, and we do around 20 to 30 passagings minimum. We maintain pluripotency, means a quality to go into mesoderm, endoderm, or ectoderm. And then we declare it that this line is holding on well with normal karyotype. We can use these cells continuously and start making heart patches or brain patches. Now, how do we do it? So once we have made a stem cell line of pluripotent stem cells, thus we write it, we give it certain initiators, and we can make either heart, most common, or make neurons. At NBRC, we had worked on creating dopaminergic neurons, and we were working on the mouse models at NBRC at Manesar, but the research is on the heart and pancreas and retina. These are the 4 places where the maximum research is going on in the world. So after we do this, this can be used as a patch. So in case somebody's got a coronary spasm and he's got damage to his heart, we can make a glue. We can make stem cell glue, and we can repair that patch by just patching it up. Same thing with neurons and pancreas for diabetes. It's very complex because it's got multiple cells. When we have a single cell is fine, but when we have multiple cells, it becomes a challenge. But still, the work is on for neurons. And for neurons, we are working on Parkinsonism. This is one way the research is going on. The second research about embryonic stem cell line is, suppose somebody has got Down syndrome or got thalassemia or some other disease, which is a genetic disease. ICM is taken from them either by induced pluripotent stem cell line or by doing an IVF. These cells are grown. So now we got colony of cells which have got abnormal karyotype, or it can have abnormal gene. The studies are done to find out how can we repair this gene. So we knock out that gene. Knockout proteins, primers are there. Those genes are known to cause a particular genetic trait. The genes can be knocked out, and then the genes are stuck together with inducers, and we find that that gene which was making an abnormal protein stops making that protein. So this is the research field where people are working, and this is what you should know than only knowing about definition of a pluripotent stem cell. So we need an unlimited supply of specific type of cells, and that's the reason that the whole world is now looking towards stem cell labs to do some magic. So this is a colony. Dr. Kuldeep gave me this lecture. I had to dig in my own old atlas to take out my pictures. This is what we had grown at Technion in Israel. This is what you see here is these are down below. These are all the feeder layer cells. So we make a plate, we coat with a feeder layer of cells, and then what you see here are the cells which are part of inner cell mass. There are a few pictures I'll show to you. They have a different color. So we coat them, and this is the way a colony is forming. This is a colony of inner cell mass which is growing over these are the human foreskin feeder cells. This is a colony which is forming, and it looks beautiful at 200 or 3000 magnification. 300 magnification. They look beautiful, and at a good magnification and good phase contrasting, you can see beautiful the way these layers are forming. So this is a feeder layer which will feed this, and we always cover it with the DMF media. This is a media which we use. We use some knockout serums, we use some MEF media, and we overlay the media with this, the plate with this, and then we keep watching this plate routinely. You'll find these are the human foreskin feeder layer cells. So what we do is that when the child is born, the foreskin is taken, foreskin is cut, crushed, multiple times we clean it, and we make this feeder layer, and it coats our plate. Or we can use a matrigel. Matrigel is a non-biological feeder layer on which we have put this colony, inner cell mass colony. Now you find that they're becoming yellow. So they are maturing now. And this is the right time of passaging. In case it becomes like this, it is maturing. And so this is not a good thing to have. This is the right time when we start trypsinizing. This is in case we missed it, or the colony was bad, this colony of inner cell mass has gone into senescence. It's become old very quickly. So nothing much can be done to this because this colony has already probably divided. And then we do trypsinization. At this point of time, we do trypsinization of this colony. There are multiple colonies which we are growing. And then we use a hemocytometer and count these pluripotent stem cells. It's a formula for counting, the way of counting it, and they convert it into millions, and then these can be grown. They are centrifuged, and then we again plate them, and we keep plating them for a few months till the time we are sure that nothing is going wrong. So like we have medias, we have single step media and sequential medias. These are the medias which we use when we are when we are working in an embryonic stem cell laboratory. This is our team which created the first. I was fortunate to work with them. They got large number of first in the world with them. They're from Haifa. So embryonic stem cells are derived from the embryos at the developmental stage before the time before implantation can occur. So when we go to a day 5 cell stage embryo, we take out the cells. As I've told you, we use laser and take out the inner cell mass. And remember that all these blastomeres has got potential to give rise to any cell of the body. So they are pluripotent, but they are still not differentiated. On day 4, day 5, they are not differentiated. So they can be taken in any direction. So the first differentiation will start from day 5. As you can see here, It'll start differentiating from day 5. So before that, we take out the ICM. You can use any method, but take out an ICM and plate them over a feeder layer plate. Like we do embryo culture, we do a whole ICM culture here, but here the plate has to be coated with a particular feeder layer, or some medias are also available on which we coat them and then they start growing. So these are the pluripotent ICM cells, and we keep growing them and not allowing them to escape and start making heart, liver, bone, neurons, blood, mesoderm or liver. We don't let them escape. We keep, before they could escape, we stop them, cut them open, and again culture them. That's the beauty of doing embryonic stem cell culture. They can proliferate for times unlimited. They give you unprecedented access to the tissues of the human body. You can grow any tissue of the human body. And the most beautiful part here, which we have to understand here, is that the moment we are ready, when we have enough colonies grown, we can always add some factors to it and make them into a particular stem cell line as we require. So basic research, they are fantastic tools for us to learn. We can do drug trials and we can do knockout gene analysis and studies on them. Degenerative diseases, they work very well. We can do transplantation of tissue after maturing them into a particular way. Now they say that in case somebody has got a damaged liver and we put his own pluripotent stem cell lines into the liver or in the bone marrow, they can self-correct and start making normal cells. Though we have not achieved much victory into this, but the day is going to happen, it's going to be a path-breaking way. Parkinsonism neurons is a big study which is going on, followed by diabetes. There are multiple challenges. The more complex the tissue will become, like bone marrow become pretty complex, neurons become pretty complex, the challenges to start, but still we are very close. And we did make a lot of neurons which could produce dopamine neurons in the mouse model. So embryonic stem cells are the best method or the only method, and this is the most important part of transplantation and translational medicine. The most perfect example of bench to bedside is human embryonic stem cell research. And I always call it is translational to transplantation and medicine. And the time is not very far off when these things will be available in the ATM. When we were talking, when we used to discuss with teams at Israel, we still talk, time will come when you'll go to an ATM, you'll put your finger there, it will scrape your finger, take your cells, and do induced IPS. So from your diploid cells of your skin, they're going to make embryonic stem cell lines. So they have got antigenicity. So they will convert it into pluripotent stem cells, and then they will make a cardiac patch, neuronic patch. So in case somebody has a diabetes, nothing to be done. Just go to the ATM sort of a equipment. It will be open machines. Give your skin sample, you just scrape your skin sample, and after 2-3 months you go and get a vial like this of your pancreatic transplants. So go to any transplant surgeon. Translation has been done by the machine. So go to any transplant surgeon. He's going to open this and inject the thing inside your pancreas or your brain, wherever you got a damage, or your heart, and things start working again for you. This is the type of dream which we have, and these are the expanded possibilities of these unique cells. Culture of embryonic stem cells, as I've told you, ideally we have been growing them on the mouse fibroblast. So what we do is that we take a mouse, we cut open the the uterus of the mouse, and then we just crush it. They've got multiple uteruses, we crush them and make a mouse fibroblast layer in the presence of bovine serum, or we can, nowadays many people are going in for matrigel or artificial layering. So they sustain the growth of the pluripotent stem cell masses. We can also add, nowadays we got leukemia inhibiting factor, LIF is added in very small nanogram values, and this will modulate through the activation of STAT3 transducers, and it won't allow, it will allow it will help them to grow them without any feeder layer. So serum free culture is not much recommended, but research is on. When we normally we are adding bovine serum to these feeder layers, so that answer we don't have, but probably time will come when we'll have everything human or recombinant and things will become better for all of us. BMPs can be added. They are bone morphogenetic factors or proteins. They are added to, they will help, they will inhibit the differentiation. So BMPs are added, LIFs are added. So they won't allow the human stem cell lines to grow into any other stem cell line. They will support the ES to grow into pluripotent stem cells line only. So once we have done the culturing, and once we have got the colonies available with us, so we have cloned the human embryonic stem cells, then in case the karyotyping is fine and the markers of pluripotency are available, then we can go in for multiple things. But just remember that when we are doing continuous embryo culturing, passaging, there can be genetic or epigenetic changes. So for every passaging we do fish, or nowadays it's doing NGS for the complete karyotyping. This is being done. So prolonged cultures can cause problem. So culturing has to be done in a very nice way so that things don't go wrong. And the most important thing, as I've told you, is making human stem cell lines through PGD. When you get a stem cell line of DMD or thalassemia or any other disease, we can grow those stem cell lines and then we can work on them and try to see how we can knock out that particular gene. And at Technion, at Haifa, they got different floors which do such knockout studies. They've got cans filled of all these PGD samples, and they keep working on them. They are a think tank for the whole world in embryonic stem cell research. So looking at the pluripotency of the ES, as I've told you, they shouldn't start dividing. They have to maintain pluripotency. So octamer 4 is one of the proteins which is always said to be a good marker. And this is our favorite, but it is not insufficient. No marker can be sufficient for this. Then another one is nanog. As I've told you, when cells start dividing, the nanog starts disappearing. So nanog and octamer 4 binding proteins are being used regularly. But as we are moving, there are many other factors, but very important factor is looking for the epigenetic changes and then looking for any karyotyping changes. They occur very quickly. In case something goes wrong in the cell, karyotyping will start getting altered. Genetic manipulation, I've already told you the way ahead. And differentiation, I've also covered that their main role will be replace or restore tissues in diabetes, heart disease, Parkinsonism, and spinal cord injury. So these are the certain things I wanted to speak to you about as far as the embryonic stem cell lines are concerned. And this is a very beautiful approach, and always an embryologist like you is part of the team, because they need somebody to cut the embryos and take out the ICM or do pronation. We call it when they use pronase. So this is a very good career option for all the young embryologists in case you want to go into these bigger studies. You can join an embryonic stem cell lab, and they got a lot of work to do, and it's very satisfying career you can achieve. When we compare mice model with the human model, mice model is always very easy to do. So what we learned when we are learning about culturing, that mice embryos are very robust. Nothing happens to them. In case even if you do some, when we do some studies on them, nothing much happens to them, and they grow pretty well. And the blastocyst conversion of a mouse embryo, of a mouse cleavage cell embryo, is very high. It's more than 80 to 90%. But that doesn't happen in humans. Human fertility is compromised, and so is the creation of pluripotent stem cells line in the humans. So that I've covered my first part of my talk. I've taken 23 minutes, but that I thought was the most important one for you to know, because this gives us a better insight into translation to transcription to transplantation medicine. So I took some more time for this. The second topic, which is very important for us, is gamete interaction till first cleavage. How does things work? How do gametes interact? What I've been hearing, what I heard, I think topic has been very well covered by my previous speakers, but I'll just try to touch upon certain points. I've been given the following headings under which I have to speak: fertilization, acrosome reaction, signaling, sperm decondensation, oocyte activation, meiosis 2, pronuclei, and spindle formation. This, I heard the conversation has already been covered pretty well. So you have to know certain things which are important, and these things are that we have an M2 metaphase to egg here, and we have got a sperm here. When they both meet, then the whole process starts. So we have fertilization occurring here on day 2, as you can see. Then we have zygote formation. We'll talk about PNs, and then we have cleavage. So I have to cover something till here. So this is I'll be covering this particular box as I go ahead in my talk. So we have to understand that we should have a clinical implication of whatever I'm going to speak. So we have heard the terms like fertilization failure or implantation failure. So there are multiple reasons of having an implantation failure. Before that, we can have fertilization failures. So these terms can be due to altered oviductal fluid components, and this commonly occurs when there is tuberculosis or hydrosalpinx. All these things, they come and they lead to failure in fertilization. So we have failure occurring at this point of time. So we have something going wrong at this point of time. There can be a defective sperm. There can be capacitation, acrosomal reaction, fallopian tube blockage, though we're not going to talk about this. So these two are the ones which lead to problem: defective sperm, capacitation, and acrosomal reaction. Implantation is not part of my talk, but since I'm on the slide, there can be altered uterine components and addition profiling and defective endometrial receptivity. But I will restrict myself to fertilization failure and not talk about this. So what actually happens? Before I go to this, I will tell you about how does the eggs get fertilized. So I'll just draw a mature oocyte which is covered with cumulus complex. And these are all oocyte cumulus complex cells are here. We have a sperm And sperm, we know that sperm has got DNA present here, and then we've got acrosome. Acrosome should be more than 40% and less than 70% of the head size. This membrane has got some receptors. They are called HAR receptors, and this is the acrosome. We have acrosome here. Acrosome contains hyaluronidase. Then there are certain proteins like pH 20 and other things which I won't talk today, but we should focus on these hyaluronan receptors. The cumulus complex is around the zona pellucida, and here we have the olema, and here we have the spindle lying scattered with no centriole. You should know that egg doesn't have a centriole. Centriole will come from the sperm. All these cells, here we have hyaluronan. So these sperms which have got receptors, they will always come and attach with the hyaluronan, will bind with the hyaluronan receptors on the sperm. The binding has occurred. This will trigger with calcium oscillations. There will be triggering and there would be release. This membrane will go, and then hyaluronidase will start dissolving these cells. This is called acrosomal reaction. When it occurs, all these cells will go. Sperm now will be able to come near the zona pellucida, but now there is no acrosome here. Acrosome has already triggered off. It's only DNA is here, and then the sperm is able to go in by a process called capacitation. So we have something very beautiful coming. That is, initially we have capacitation. This can occur in the media. This can occur in the female vagina, occur in the cervix. Second step is we have acrosomal reaction. Third step is penetration. And the fourth important step is sperm decondensation. Dr. Kuldeep has given me these topics to cover. So I'm just covering them one by one. I've come to sperm decondensation now. So what is sperm decondensation? You have to know that the DNA of these sperms is always having quaternary binding. And this binding is on itself. It makes it the most compactly packed DNA in the human body. This binding is all around some protein chains which change later on. Initially they are binded upon them like this. All these protein, DNA is binding on this like this, with zinc molecules coming here and there. These are histones. And as we move towards this, we get protamine. And protamines P1 and then protamines 2. So good binding means that from histones, we have protamine 1 and protamine 2 condensation in the final stages. And finally, we have more of P2. So DNA is very tightly packed. So when, at this point of time, when the sperm goes inside, the head goes inside, this DNA is highly condensed DNA. This DNA will have to open up and the chromosomes have to align. This is called decondensation. So I've told you about capacitation. I've told you about acrosomal reaction. I've told you about decondensation. And now there would be the stage where both the cells, the centriole from the sperm is going to divide here. Centriole will divide here. This part of centriole will go here, and then all these chromosomes will be aligned on the spindle, and then there's a membrane formation occurring around it. This will initially there will be two pronucleus. They will face each other like this with the nuclear precursor bodies. They fit in like this. They rub each other, touch each other, and then finally they will merge. That's we got one cell there, and within few hours it'll cleave and it'll have a cleavage situation will arise. So what happens? Few figures I'll show to you for easy understanding. Sperm binds to the zona pellucida. This is stage number 1. Then, as you can see, there's a plasma membrane here. There's a zona pellucida. Then 2, binding has occurred. And for this, there's a very beautiful test called high urinone binding assay, HBA assay. The slides are available in the market, and we should have minimum HBA binding of more than 70 to 80%. In case it is less than that, there can be total fertilization failure. In all the cases where we have total fertilization failure, we should always do HBA binding. So the contents are released, and then we have acrosomal reaction. With this, all these cells will get dissolved. Sperm will go. The hydrolytic enzymes burrow through the zona pellucida and enables binding and fusion of the sperm with the egg. So now the acrosome, this acrosome, as you can see here, this blue thing was the acrosome. Acrosome is releasing hyaluronidase. It is releasing here also, and finally we'll only have green DNA. There is no hyaluronidase acrosome here, and then this egg nucleus, and then finally this will go inside. The sperm nucleus enters the cytoplasm. This is the process of decondensation. So a highly condensed DNA, which is bound on a protamine proteins like this, zinc everywhere, interwoven, it will go and starts opening up. It start opening up. Decondensation, like what we do in the halo test. We do SCD testing or we do NCD staining. It will open, and this is what we'll get. From this small one, we get decondensation, and then we get a bigger one, and this forms a PN. So we got a male PN and we got a female PN. They both look at each other, and then those anaphase and chromosomes start dividing, coming in contact. There is some amount of overlapping, and then the thing goes on. One term I wanted to introduce again for you. I have told you the term called acrosome reaction. This I've covered for you. I've covered the term decondensation for you. I've covered the term capacitation for you. One very important term is cortical reaction. Cortical reaction was not given to me to speak on, but I just added cortical reaction here. After the sperm goes in, then the reaction will occur with the cortex, and the whole thing is going to close so that there is no polyspermy. So cortical reaction is a very important step, and it occurs the moment the sperm head goes inside the cytoplasm. So when you look at how the fertilization occurs, we have spoken about meiotic release of the first polar body. Then we've spoken about the sperm nucleus going inside, decondensation. We have ovum nucleus, male sperm nucleus, polar bodies. There are two polar bodies are seen. Then meiotic spindles are formed. Chromosomes are aligned. Centriole comes from the male, divides into two. And then we find that this is what is happening. All the chromosomes are getting aligned like this. This after the pronucleus is formed and syngamy is occurring. And then syngamy will occur, and then we'll find chromosomes will align at the equator. Anaphase of the first cleavage division occurs now, and then break comes and we got cleavage. This was given to me to speak to, so just covered in short about how it happens. And this part, I've already told you that most important thing you have to all know as an embryologist is that there's something called hyaluronan receptors and binding of the sperms with these receptors to the hyaluronan containing cells in the OCC. They are the game changers. Fertilization is pure English, but as a embryologist or as a clinician, we have to know that this can actually go wrong. So whenever we are having a total FERT failure, always think that something may be going wrong and there may be receptor problem also. But in today's world, we can fail for the first time, but not for the second time. So in case we fail, we have TFR, next time go for an ICSI. And in case no ICSI, do an HB binding assay, only then we move ahead. So time is 6:39. I'll just take few more minutes and cover my other two topics. So coming to the fetal ovary. The fetal ovary, how the things work. As we have said that we have formation of a zygote. This is the male and this is the female. In the female, there is no SRY gene. In the male, there is an SRY gene. So after some time, when the embryonic gonads are forming, in case there is SRY gene, the whole thing will move towards formation of testes. When SRY gene is not there, then we get ovaries. It all depends upon the presence or absence of an SRY gene. We get testes or we get ovaries. Till the time embryonic gonads are not formed, they don't know what they're gonna become. They'll become male or a female, they don't know. But depending upon the presence of SRY gene on the Y chromosome, things change. So for your exams, you should know that this is an dividing force, or it is an influencer. We call it, this is the main influencer or driver for a particular, to move them in a particular direction. Now, what is an SRY gene? This is sex region Y gene, and it is found only on Y chromosome. So you can see that it is present, the gene is present on the P arm of the Y chromosome. And in case this gene is present, then the embryonic gonads will get direction towards making testes. In case they're absent, then probably we'll get a female. SRY genes are correlated with the presence of testicular determination factor. In case TDF is present, then we get testes. If no TDF, we get ovaries. And then here comes MIF. In case there is present of inhibitory factor, then we get male genitalia. And in case we have no TDF and no MIF, then the Mullerian ducts will make female genitalia. So you have to know about TDF, MIF, and SRY gene. They commonly come in the exam as an MCQ. This you have to know. So fetal ovary is the beginning of the ovarian development. So till around, it starts from indifferent gonads initially. As you can see here, the genital ridge forms near the dorsal mesentery. At 4 weeks, we find that there's a proliferating epithelial cells and this mixture of somatic and germ cells. Still, we're not clear what's going to happen. When we have an ovarian development, we'll find that changes occur in the surface epithelium and they become the surface epithelium of the ovary. In the male, we'll have formation of tunic albuginea. In the female, we'll have anatomy of the female ovary coming and ovarian cortex is developing, surface epithelium is there, and we get oogoniums and primordial germ cells come here. And in the males, we get tunic albuginea, the whole assembly which we know, which we actually are aware of, everything forms, and by 16 weeks, starts at 8 weeks, and by 16 weeks, we know what we are dealing with. So sexual differentiation starts around 7 weeks of intrauterine life, and it ends at around 16 to 20 weeks. So initially we have migration of the germ cells into the ovary, formation of the follicles, and distinction of the ovary into cortex and medulla. This is a formation of the gonadal ridge from something called gonadal ridge epithelium-like cells. They're called Grell cells. They play an important role in formation of a human ovary. So ovary commences at the mesonephric surface. This is the mesonephric surface. Here, the yellow cells will start multiplying. As you can see here, they'll start developing and ovarian development will start. Then I have told you about the Grell cells, which are gonadal ridge epithelial-like cells, which come into play, and then you'll find that it starts developing into various parts of the ovary. We got granular cells, we got capillaries, somatic apparatus, primordial germ cells, oocyte, oogonia, basal lamina. So everything starts at the mesonephric surface epithelium from these yellow cells, and then finally it will start multiplying and dividing, and we get the surface epithelium. So we get ovarian cortex here, that is surface epithelium, and just below it we will get the primordial follicles or germ cells, which are primitive oogonia, and they form the basal layer of the ovarian cortex, the most important layer of ovarian cortex, and all below will probably be having medulla. So this is the surface layer containing primordial follicles. They all contain primordial follicles, and this is what we have here is the stroma or the medulla. This structure would have been covered for you already, so I won't touch upon that. This is a typical germ cell in a female, and I think we all know about it, so I'll skip it. So the migration of the primordial germ cells to the ovary is an important aspect that starts around 3 to 4 weeks. Primordial germ cells are present somewhere near the hindgut, as you can see here, and they are migrating. They migrate to the midgut, and they finally form as they're moving up, gonads are forming, and they form the gonadal ridge. See here, they are moving up from the hindgut, they move up, and finally they go and form the gonads. This will be the gonadal ridge which is forming. So along with the oogonia formation, we have formation of the medulla, and after the medulla has formed, That's the time when the ovary is completely formed and it starts. At that point of time, all the primordial follicles are the maximum in number in the ovary, and it is now a self-sufficient organ. This is a similar picture. We show that primordial cells are migrating from the yolk sac to the primary sex cord. I think we had a small discussion about this some time back. So primordial follicles, they migrate from the yolk sac and they form the sex cord here. These are all the primordial follicles. This is the cortex, and here we have the medulla. And that's the way how things are working. So we look at around in the early, initially we have migration, then we have bipotent gonads, then we have sex determination. This is a time when we have a play of TDF, SRY, or MIF. These three things come together, and then we get either a male or a female. If we get a female ovary, then that's the way the things move. Initially, we have primordial follicles, immature follicles, and finally, as you would be learning, the cells will start multiplying. And so, from the journey which starts at 5 weeks, it ends at menopause. So, for an embryologist, this was all, or for any clinician, this was all some amount of English, which actually doesn't have any relevance as far as our clinical management is concerned. But more important is the journey which starts at around 4 weeks for us. This journey will continue, will culminate into an ovary at around 16 weeks. At 20 weeks, when we look, we got nearly 6 to 8 million eggs. At birth, 1 to 2 million eggs. Puberty, we have got around 3 lakh. 3 lakh eggs we have at puberty. And then at 35 years, around 25,000, and at menopause, 1,000. So what starts at a very beautiful journey at around 4 weeks, with the absence of TDF and SRY and presence and absence of MIF, the journey peaks at 20 weeks and finally culminates into menopause, where we may not probably be having any cells at all, and the lady becomes menopausal. So then embryologists, you must know this clinical correlation, when to intervene. So in case you understand everything, then you'll know that which all patients can have poor quality oocytes, in which patient we can do egg donation or embryo donation, whatever we do for to help the patient, indications. So you have to correlate the journey of a female, a girl who's come to you for treatment when she was in utero at 20 weeks till she goes into menopause and she comes to you. So her decreasing fertility, I think you have to know, and then correlate. You know that she started with around 4 lakhs at puberty, and now she's barely 20,000. There is a fall in her egg reserve, and in case we don't act here, it'll probably go and then it will disappear. There will be end of fertility and menopause. Excuse me. Fetal testes, as I've told you, we have a genital ridge, germ cells from the yolk sac. It's a bipotent gonad till the time SRY comes, and then there are SOX9 and DAX1 genes come into play, this upregulation, and then we have formation of testes. Wolffian duct stabilizes and Mullerian duct regresses. Testes are formed, they descend, and external genitalia are formed. That's the way the things move. And for the exam, you have to know these things. These come in the exams quite a number of times. You have to know that which genes binds with SRY genes and upregulate SOX9, which genes are regulating MIF, which gene SF1 is important for testosterone production, These few things may come in the exam, which you have to know. Otherwise, as an embryologist, you may not be using this knowledge to daily application. To sum it up, I've got 8 more minutes. We have early indifferent stage, SRY, TDF, MIF, SOX9, DAX1 comes. We have testes from 7 weeks onwards, ovary from 8 weeks onwards. Wolffian duct develops into testes. Mullerian ducts degenerate in the male. And that's the way the gonads are formed. This is again the figure which I've showed you. Initially, we have migration of the cells. They come to the gonadal ridge, and under the effect of the hormones or some genes upregulation, we have formation of the male testes. And the male testes here, they descend down. In the female, they stay in their position, but in the female, there's a migration. In the male, it's a migration. So the testes, at the 7th month, they reach the internal inguinal ring, and finally it comes down. So primary descent of the testes, they start at 5 weeks. In the female, there is no descent of the ovaries. In the male, there is a descent of the testes. This you have to know. Well, here I end my talk. In 55 minutes, I've taken you through a small journey where I've spoken to you about embryonic stem cells line. I've spoken to you about various things which happen during fertilization. We've spoken about capacitation, we've spoken about acrosome reaction, we've spoken about cortical changes, we have spoken about decondensation, we've spoken about chromosomal arrangement, and then we've spoken about cleavage. In short, in around 20 minutes, I've spoken about the formation of female and male genital organs. I've told you what all is important for your MCQ exams, which may come. I've told you what is important for the exam and what has got clinical relevance. So with this, I will end my talk here. I'm sure that you'll be able to apply some of the things which I've told you to your daily clinical practice, and you'll all become very successful embryologists, and you'll all will qualify your embryology exam also. Kuldeep sir, Jayant sir, Arne sir, I've ended. Thank you very much.
Thank you very much, Dr. Pankaj. Sir, it was interesting and a very good journey for all the delegates to learn all the various different aspects. If you have a few minutes, I'm sure there would be some questions that the candidates would like to ask you, if you're okay with that, sir.
I'm totally free, sir.
Okay. Arne, would you like to come in?
Yes, there's one question for you, Pankaj. Can you please elaborate more on capacitation? That's from one of the
capacitation is a process. Capacitation is a process which capacitates a sperm to undergo the further changes. Capacitation is a process which will clean the sperm. So how does capacitation occur, and what is the relevance of capacitation? Capacitation, after the sperm is ejaculated, sperm has to be activated. Sperm has to be activated. Activation can occur in the multiple ways. The activation or capacitation can occur in the female genital tract. It can occur after the ejaculation. It can occur in the cervix. It can occur when the sperm is moving up. In our parlance, when we are working in the laboratory, there is no cervix, there is no vagina. Then how does the sperms capacitate? Sperms capacitate by multiple methods. In the laboratory, they can get capacitated or they get activated to undergo the maturity of the receptors, the capacity to undergo acrosomal reaction, the capacity to initiate cortical reaction, and the capacity to undergo decondensation. This occurs either when we do the sum up of the semen sample. So in case we take some semen sample, just do the plain simple sum up, or we do double density or a single density, or we wash. Just remember, the very touch of the sperm with the media which has got milliosmolarity, which has got the same quality of human tubal fluid. So any media which has got adequate amount of glucose, non-essential amino acids, and proteins, which is akin to the human tubal fluid, the contact of that media with the sperm can lead to the capacitation. So capacitation occurs this way, and why is it important? I have told you. Dr. Arne, this is what I like to speak about capacitation.
Jayant or Kuldeep?
Yes, there's another question that has been that needs answering, sir. And
yes,
the question is, can you please give some stress some importance on the possible genes which are responsible for male and female gonads?
Sir, I've told you there are 3 genes which are important. These 3 genes, I guess I can go to my presentation is locked now somehow. So there are 3 genes which are important. There is a, excuse me, I got a bad cough, so kindly excuse me for that. We have got a gene on short arm of Y chromosome. P arm of Y chromosome, short arm Y chromosome. That gene is called SRY gene. That gene, in case it is present, then the primitive gonads will convert towards the male anatomy and the male descent of the testes. Then they have got testicular determination factor, TDF. When the TDF is there, testes will be formed. And third gene is MIF, that is called Mullerian inhibiting factor. So when anything inhibits Mullerian agenesis, in the female we have Mullerian tract, in the female, and the male we have got a Wolffian tract. So in case we have got a gene which is upregulated and which leads to degeneration of the Mullerian gene, Mullerian apparatus, that will lead to formation of male again. So we look at MIF gene, SRY gene, and TDF gene. Then, as I've shown you some figure, that at some point of time there is upregulation of certain genes which come up, and these genes lead to these genes like SOX genes, and this one wanted to show to you. These genes are DAX1 genes and SOX9 genes. They play an important role for the male genital tract development. Out of these, the SF1 gene, this one gene which is upregulated, that is called SF1 gene, that is important for testosterone production. And then the SF1 gene is the one which binds SRY gene and upregulates SOX9 genes. So in case you ask me, for an exam purposes, what are the things which are important for our understanding also, why exam? We have TDF, MIF, SRY, SOX9, SF1, and then we have DAX1. They are the ones which help in various stages in stabilizing testicular germ cell production, stabilizing the Wolffian duct and Mullerian duct agenesis in the male. And their absence will lead to formation of the female genital tract. And these genes also help in testicular descent, which doesn't occur in a female patient. Dr. Jayant have ended my answer.
Sir, I've got two questions for Dr. Ruin. The question is there any role of HCG injection in stimulation of follicles instead of LH?
So many people do use low dose HCG instead of LH, but routinely we are not using. We prefer using the LH component, but you can use low dose HCG for LH. It has the same action.
Okay. And final question to Dr. Pankaj sir. The question is, if a patient is undergoing IVF and they have surplus blastocysts, if they do not store umbilical cord in future, can they use the blastocysts to treat any problems?
Why not, sir? I actually am totally against umbilical cord banking. Is not the right thing I like to talk about. It's very controversial, and there are no guidelines about that. We are only doing it for commercial reasons. So IVF, umbilical cord blood banking, UCB, I'm not. I'm dead against that. But yes, she's right. In case we have surplus, see, all these cells have got massive antigenicity, because once you make a pluripotent stem cell line, they are antigenic. They are actually antigenic. And when we want to create a cardiac patch or a pancreatic patch, this can be translated, but cannot be transplanted into the third person. So we have to develop these cell lines for that particular person itself. So I fully agree with you, Sally. In case somebody feels that he's going to have Parkinsonism or some disease, definitely 1 or 2 blasts can be frozen, and then they can be converted into the pluripotent stem cell lines. No problem. But what I told you now, the way things are going on, it's not around, maximum I say is around 5 to 7 years, you'll have those ATM machines where you'll be getting your skin punctured, and they'll give you your own cell lines. But yes, why not? Logically, you are correct. This can be done. But now is the era of induced pluripotent stem cell lines, where we reverse from our skin cells or body cells and then create pluripotent stem cells line in reverse way. That's the way we are going now.
Thank you very much, Dr. Pankaj sir. I would now like to ask Dr. Kuldeep to wind up the session today, and also like to thank all the speakers for this session. All the delegates have very much appreciated your presentations, and they've taken a lot of information, and I very much hope that if they have any more questions, if they can contact you directly to answer those questions, and you would be happy to do that. One of the things that the candidates have requested last time and this time as well is if you could provide them with the reading material pertinent to your lecture, then at least they can go back and read that those material and revise for the examination purposes. So once again, thank you very much to all of you. Word from Arne, and then winding down from Dr. Kuldeep sir.
Oh, I just want to say I enjoyed this funny morning and few afternoon, and hope to see you next week.
Thank you to all speakers. I think today's lecture, they were really very, very good. Kesab, Surveen, Sweta, and Dr. Pankaj. I hope everyone has got a lot of stimulation to read more and more to prepare for exam. There are a lot of questions many people are asking about whether they will get the presentation of the lectures or not. So for that we have decided, this is just an announcement, that we will be uploading all recordings of all lectures for next 1 month once we finish the course, they will be available for all registered delegates on IFS website. Of course, it will be password protected, and this will be available only for 1 month. So in the month of December, you will be having all recordings available for 1 month, and then they will be removed from the website. So you will get the password once we finish the course, and then that you can. So if you have missed some part or some lecture, you can again go on the website and enjoy the lecture. With this, I would like to thank all delegates as well as the faculty, Dr. Arne, Dr. Jayant, for staying throughout today. And I am hoping that next Sunday we will again meet at 3 o'clock. Thank you very much.
Thank you. Thank you.
Thank you.
Yes.
And goodbye to everyone. Stay safe until next time. Thank you. Bye-bye.
And all the best for your exams.
Thank you.
Bye.